worked into the different direction, like some different analyte we have to this monitored, like the cholesterol, triglyceride, glucose, urea, and some other analytes like in the form of antigens, like the pathogen detections and some DNA biosensor. So based on their our analyte requirements, we can choose biorecognition elements. And after this making this hemobilization of these biomolecules onto the biocompatible layer, that we can link this electrode with your transducers. So transducers that depends on their biochemical reaction which are going onto the electrode surface. So we can choose if there is some electrochemical reactions, like electrons will be generated, or it's some change in their potential, change in the current value, we can choose the electrochemical transducer. If there is some optical change, the change in their angle, change in the like the SPR based and this biosensor we can develop. So based on their requirement of their biochemical reactions, and based on their this facility we have in our lab, we can make the biosensor. So after this transducer these selections that we can attach with the amplifier to enhance their signal, and finally we can monitor their output. So on the basis of their different type of the transducer, like piezoelectricals, electrochemicals, optical based transducer which can be used. So in case of the electrochemicals, there is a 3 electrode system mostly are being using, that consisting of the working electrode, counter electrode, and the reference electrode. So 3 electrodes mostly is using, and these electrodes to have we will put into the electrolyte solution, and their response change in their current value, yeah, potential control we can do during their analysis. So in the there are the different type of the electrochemicals techniques are available to read out their output in the form of the current, in the form of the potential. So in the form of the current, mostly DPV, differential pulse voltammetric, cyclic voltammetric, and in case of the impedance, like impedance, the RCT, charge transfer resistance also can be monitored. In case of optical biosensor, there is a some like in the similar way, different way that we can read out their signal. The color will be changed if you are adding some analyte, and there is a biochemical reaction will be take place. So color will be formed. Or second, like SPR, surface plasmon resonance weight biosensor also can be fabricated. Here you can see at the electrode surface, there is a surface plasmons wave are generated, and these wave are generated that give transmitter of energy that will be 100% transmission, and their change in the this intensity after the binding of the biomolecules with the analyte, their angle will be changed. So change in the angle that is correspond to change in their analyte concentration in the form of the change in the intensity. So we can monitor their response in the form of the time versus change in their angles. So this the similar type of the curve we will obtained during the SPR measurements. But in case of the piezoelectric biosensor, here we can see there is some there if there is some physical this mass is changes onto the electrode surface, their signal will be changed. So here we can check change in the frequency we can monitor. But in my group recently we are working on the only electrochemical biosensor. Why we are using electrochemical? Because they are very sensitive techniques, and then we can analyze we can monitor analyte concentration in the form of the femtomole and in the picogram level. So and other scientists also using the electrochemical biosensor. So on the basis of our analyte, basis of our requirement, there is a wide application of the biosensor that can be used in the different area, like in the biomedical area, in agriculture, military, environmental monitoring, and the industry also. So during the bioprocessing their monitoring, we can find out that how this your sample is showing the parameter after the processing and the before the processing. Environmental monitoring we can use for some like pesticide detection, pesticide based biosensor, or some type of the pathogens which are present into the environment, like the COVID-19 is also possible to find out their presence into the environment sample as well as in the water samples using the biosensor. And some group are working for the COVID-19 detections also. In medicals, there is a glucose biosensor is a very well known at is a for like the glucose biosensor in the medical uses, and simultaneously like cholesterol, urea, uric acid, triglycerides, and some like cancer biomarkers. The work is going on. So they own the choice, and then we can make the biosensor. Now what is the advantage is why it's a more this important as compared to the conventional technique which we are using, like SPLC, like ELISA for biomedical application, because these the ELISA and SPLC, they are giving specific results, but they are expensive and they are taking more time. So to overcome these advantage this advantage of the conventional techniques by sensor that give the direct small that give the direct small this amount of sample is required, only we need one one micro ml of the volume, give the different concentration of the analyte, and also give the accurate results. That depends on our biorecognition elements which are present into the sample. So this
Madam, it's not audible. Hello? Dr. Pratima? Dr. Pratima? Hello? Dr. Pratima? All please wait. The connection will be reconnecting again. Please wait.
Yes, sir. Yeah, I connected, sir. Yeah, yes.
Now it's visible, madam. Hello, madam. Hello, Pratima. You please unmute. Hello, Pratima. Please unmute. Hello, Pratima.
Yes, sir. Yeah.
Yeah, please unmute. And yeah, you can start
O-
now. You can
Okay,
share your
sir.
screen.
Yeah, yeah,
Yeah.
yeah. Yeah. So these are the wide applications of biosensor that can be used for fabrication of the different biosensor. Now, advantages also that I have discussed, and this is some the first biosensor electrochemical biosensor which are fabricated in 1962 by the Professor Clark for the glucose device, and I think everyone are aware about the glucose biosens which is the commercially available. So this how we are using this we are making the biosensor for electrochemical biosensor that consisting of the transducer element that convert the recognition elements signal after the interaction with the analyte, and then
वाईफाई कमिट वाला तो नहीं कर रखा, कमिट वाला तो चल ही नहीं रहा। हाँ कनेक्टेड चल रहा है।
Hello, sir. Hello. Hello, sir.
Okay, madam. We
Yes,
can
sir.
able to hear.
Yeah. Yes. Yeah. Hmm.
Okay.
Hmm.
Please share your slides. Okay, coming.
Hmm.
Yeah.
Yes, sir. Sorry that there's a internet problem from both side. Hmm.
Okay, continue,
So the
please.
yeah, now that this is showing that why we are using nanomaterials for the development of the biosensor, because you know that provide the high specific area, and that the high specific surface area that help us to binding of the more this biomolecules that they provide this high sensitivity. And these most of these nanoparticles which we are using, these are the biocompatible, like the carbon based materials, like metal oxide based nanomaterials, and these have showing the high chemical stability. They are not react with this direct with the analyte and the other, like the pH and the pH effect or electrolyte solution that not showing their the chemical their behavior changing. So some of these nanomaterials like the semiconductors and the like the RGO and the this RGO, they are electrochemical activity active. So electrochemical active by nanomaterials, they provide also help to provide the higher sensitivity, and due to their high absorption capacity of these nanomaterials, that also increase the stability as well as their the sensitivity of a biosensor. Because some of these the parameters like we have to they consider in mind that these nanomaterials that help to provide the wide detection range of the analyte, and their sensitivity also increase, their stability also increase. So before going to choose their nanomaterials, we have to think the which nanomaterial will be the more suitable, which keep all these the biosensing parameter in the like improved biosensing parameter. So nowadays, you know that if we are using nanomaterials, that some due to their high surface charge, the nanomaterials they are aggregated very fast during the synthesis also. So if we this modified their surface with some type of this a capping agent, some type of like molecules like amino acids or some polymers, their stability can be increased. Their the property of nanomaterials can be maintained. So there some surface functionalization is a best way to synthesize the nanomaterials, and that can be also provide a more functional group which help to bind the biomolecules, also biomolecules for the biosensor application. And these nanomaterials, like some of these nanomaterials, are the toxic in nature. If we their surface will be linked with some biocompatible material, their toxicity can be reduced. So keeping these points in the view, the surface functionalization of nanomaterials is also the very essential. So in this direction, that different molecules like the C-tab, PG derivative, copolymer, and the silica, and like the polypeptides, some DNA. So these are the materials are being used for surface functionalization. Some of these like the citric acid, C-tabs, and the PG molecules we have explored in our research group also, and they are effectively enhance the sensitivity as well the stability of the nanomaterials. But how we can make this bioconjugation? So there is a some type of like binding the chemistry is involved, like the covalent bonding we can use there, that like amide bond formation, imine group formation, and the disulfide bridges between the two molecules, they help to their covalent interaction of the molecules. They provide the stability of nanomaterials with the biomolecules. So, some of the nanomaterials, they have their high isoelectric points, like the zinc oxide or cerium oxide. If we use some molecules that having the positive charge, the nanomaterials and the positive charge, their isoelectric quantity is very high, like the zinc oxide, that they are showing the positive charge, and then we can use some this the negative charge molecules, other polymers. So due to their electrostatic interaction and some hydrophobic interaction, these molecules provide their binding. So on the basis of these chemistry, like the surface group onto the nanomaterials and their functional group onto the second molecules which are going to functionalize these nanomaterials, which can be planned their chemistry. And there is some biological interactions is also available in the literature. They are providing the very strong bond, like the antigen antibody, Watson Crick pairs, and the avidin biotin. Avidin and biotin is a very known this chemistry that people are using for binding of the molecules. So in our group, like we have fabricated different type of nanomaterials, like the iron oxide, lanthanum hydroxide, vanadium sulfide, carbon dots, zirconium oxide. And continuously we used these functional group, like the citric acid, oxalic acid, L-cysteine. Mostly this L-cysteine, these molecules we have used for almost every this nanomaterials we have synthesized in our lab, because L-cysteine is again is the amino acid that is a very 100% biocompatible, and they also provide the amine and the carboxy group that help to bind with the other molecules, the biomolecules like antigen and the enzymes. And carbon dots functionalize zirconium oxide also we have fabricated carbon dots. These are again that 100% biocompatible. So this slide shows that which in area we are working. Like we in the JNU, after the joining of the JNU, I have started this direction, like the vitamin D detection and this cancer biomarker detections, or this mycotoxin or mycotoxin detection into these food samples also. And we have synthesized these cerium oxide, zirconium oxides, lanthanum oxide, gadolinium oxide, lanthanum hydroxide nanoparticles, and explored their electrochemical response. And these are the molecules we have used for the binding of their functionalization of these nanomaterials. Simultaneously, we are also have published some work on the fabrication of the nanofibers using polyacrylate nanofibers. And these nanofibers we have prepared using the electrospinning machine, and we also embedded the nanoparticles into the polymers. They provide again their stability of these fibers. Now, today, as I discussed that application is in the biomedical area, I will discuss the cancer biomarkers. Cancer biomarkers, specifically, we have chosen some biomarkers like the IL-8, interleukin 8, and the cytokinin fragment 21-1, CIPRA21. But there are other biomarkers also reported into the literature, which are being used for the cancer based diagnosis. So if we choose the biomarkers based diagnosis that help to predict the prevention measurement that could be formulated. And at the time of we know that the concentration of the biomarkers into saliva in the serum samples, that we can predict that which type of the disease and which type of this treatment to be required. So using this electrochemical biosensor, the main this advantages of this sample sensor is that electrochemical biosensor can be detect very low concentration that will be used for the early diagnosis of the cancer biomarkers. So in we have think to work in this direction. So we have first we have choose like the CIPRA21 biomarkers. Their concentration is in the oral cancer patients, that is 17.46, and in the normal sample their cutoff value is 3.8 nanogram per ml. So these CIPRA21 biomarker that is present into the saliva, we did some experiments, and there we got very good results in the saliva sample as compared to the serum sample. So we have selected the CIPRA21 biomarkers into the patient sample. So for this, first we have prepared some biosensing electrode, that how we can monitor their response. So this is a scheme that's showing that how we can fabricate biosensing electrode. First, we are making like nano materials. This is showing the nanomaterials like the lanthanum hydroxide nanomaterial we have synthesized using their hydrothermal the procedure, and simultaneously these nanoparticles we have functionalized with the L-cysteine. So this is the in center scheme that's showing that how the carboxy group of L-cysteine bind with the lanthanum hydroxide nanoparticles, and their terminal group of these nanoparticles OH group, that is the free OH, and the amine group of these molecules is the free that can be used for binding with the antibodies. You can see once we have functionalized these nanomaterials with the L-cysteine, and their amine group mostly is free, that using the EPD, the electrophoretic techniques we have to use, which provide at a particular voltage these nanomaterials is deposited onto their electrode surface. Here we have used ITO, that is the indium tin oxide substrate, that is a conducting. One more important point, that for the electrochemical biosensor we need conducting substrate for the deposition of nanomaterials, making the sensing area. Otherwise their signal will be not generated if we use some insulated material. So we have to keep in mind that we to use the conducting substrate for deposition of the nanomaterials. Once that electrode was fabricated, that in antibodies was immobilized using EDC NHS chemistry. So in this case, their carboxy group of this antibody surface that is bind with the amine group of this nanoparticles, functionalized nanoparticles, and there is a electrostatic interaction will be take place onto the electrode surface. So simultaneously, there are some possibility that this all the nanoparticles, they will not participate to bind with the antibodies. uh, so there is a chance that there is some noise signal, the background signal will become during the measurement. So to avoid this the background signal, that electrode has been modified with the BSA, that is the bovine serum albumin, to avoid their background signal, and then electrodes were treated with the analyte is the CIPRA21, and their response was monitored using the electrochemical techniques. So this is a way that how we are developing the biosensor electrode. So as we first going to fabricate this electrode, we have to characterize all these nanomaterials like TM, SCM, XRD, and electrochemical techniques. So in this case, first we have used the XRD. This is showing XRD results of lanthanum hydroxide nanoparticles. So this the star point into XRD, that is representing the presence of the L-cysteine into the nanomaterials, and this the prominent peak that is corresponding to the lanthanum hydroxide nanoparticles, and the particle size we have calculated using the Serrer formula, that size was found at the 18 nanometer. So this is the first way that characterize the presence of L-cysteine onto the lanthanum hydroxide nanoparticles. Then further we have confirmed their crystallinity and their shape of nanoparticles, the TM. The transmission electron microscopy study has been used. You can see that the nanoparticles, they are not much, there is a monodisperse, uh, but this figure B, that's showing their size of nanoparticles we obtained around 45 nanometers, and there onto the nanomaterials there is a white the lines white the shaded area that indicate that your nanoparticles has been modified with the L-cysteine. And we have there we try to find out how much there this the white line onto the nanoparticles, that is found around 3.5 nanometer, this area that indicate the L-cysteine is thickness is 3.5 nanometers. So we also simultaneously we have checked their d spacing value, that is correspond to the XRD data of 1110 plane, and it's confirmed that both these XRD and the PM value corresponding this like the 1110 plane. So for further functionalization of nanomaterials onto the electrode surface, during the binding of the antibodies and the binding of the BSA onto the electrode surface, we have to check that there is a change in the functional group or not. That is again the FTR study we have this done, and to find out, you can see in the first spectra A, it's correspond to the L-cysteine, the only bare L-cysteine. So all the peak that corresponding to the amine and the carboxy group, they are present into the L-cysteine, and 942 and the 5550. These two peaks are the corresponding to the sulfur group, which is present into the L-cysteine. So as the functional group, this electrode we have modified their peak, which are corresponding to sulfur group is disappear into once we have interact this lanthanum hydroxide nanoparticle with the L-cysteine. So it's indicate that there is a interaction between the L-cysteine and the lanthanum hydroxide nanoparticle take place. Now this electrode we use for immobilization of the antibodies, that this spectra 3C showing the peak corresponding to amine group that correspond to 16, 20 and the 1565. They are specific the amine group which are present into the antibodies, and in the similar way in the BSA also hydroxy group also present into the antibody as well the this a BSA. So this is the way that we are doing work for finding modification of the electrodes with the antibodies and the BSA. Now for the sensor application we are making the electrochemical sensor. So again here are the some different parameter we have to check, like the effect of the pH. What is, because you know that molecules they are changing their orientation, changing and their functional group, like functional group need the charge onto the charge as your pH will be changed. So we have to study that at which pH this electrode is showing the highest their response, highest their current value. So you can see here that different pH value we have taken, like 6 to 8.5, and the maximum the current value is obtained at around the 7 at 7 pH. So 7 pH we have selected for further electrochemical studies. And one more point, step to step modification of electrode, we have tested their electrochemical response. All these experiments we have done into the redox molecules which we have added into the electrolyte, that is FeCN6, that is the redox species they giving their signal. And this curve is the differential pulse automatic, this is a DPV curve of our electrodes. So again that is very necessary to find out their electrode modification step to step. Like first curve is that corresponding to the I2 electrodes, the second is with your nanomaterials. So you can see here there is a drastic changes in the current value. In the I2 surface the current is very high, but as we added the nanomaterials, that current is decreasing. So I2 is very conducting, that is a bare, and nanomaterials is a lanthanum hydroxide, that is a semiconductor in nature. So their current is decreasing. So again that we have added the antibodies and the electrode surface, that is current is increasing. These changes that depends on their depends on their flow of electrons into the electrolytes, because in there is a some like electrochemical this interaction of free group onto the electrode surface, because in antibodies also having the amine group, carboxy group, they participate interaction with this redox species into electrolytes. There is a not any confirm like a sequence that in always your current will be increased with the antibodies. Sometimes their current is decreasing. That depends on their this orientation of the antibodies, how they are immobilized onto the electrode surface. Because sometimes we are using the only the physical adsorption, that they are block their flow of electrons onto the electrode surface. So proper binding, the proper way of the orientation of antibodies onto the electrode surface is very necessary for their immobilization. So then again, this current is decreasing with the BSA, because there is maybe some noise signals in during the antibodies also. There are the different factors. And one more thing, once we are adding the antibodies on the electrode surface, their isoelectric points of the electrode will be changed. And after the BSA, that isoelectric point of electrode again that will be changed. So these are the factors where they affect their electrochemical response. So finally, then we find out the response time, that how much our electrode gave the response with the analyte concentration. You can see different time we have chosen with the same concentration, like 0 to the 10 minutes. 10 minutes tak we have optimized the response, and the maximum you can see that the current is not changes after the 5 minutes. Huh? You can that is a stable. So it's indicate that 5 minutes are the sufficient time for the binding of the antibodies with the analyte. So this graph C is showing the different concentration of the analyte onto the electrode surface we are added, and the giving the incubation times of the 5 minutes. And then we have checked the response with the different concentration, that the values is varies from a 0 nanogram. Zero means that without any concentration, only the bare BSA electrodes response, and then up to the 10 nanogram per ml, how it the response is changing. You can see that continuous their current value is decreasing. It indicates that that antibodies onto the electrode surface, they are binding with the analyte with the step to step, because in the low concentration, analyte concentration is very less. As you increase the concentration, their analyte concentration will be more, and the more analyte molecules will be bind with the antibody onto the electrode surface, or they inhibit their flow of electrons. So finally that current will be decreasing. So this is the curve you can see here in the graph C. The current continuously the decreasing, and up to the lower value of the sensor detection limit is that is a 0.001 nanogram per ml, and up to the 10.2 nanogram per ml. So our physiological range of parameter that is in between, that is a 3.8. So your the sensor can be detected beyond the physiological range of our CIPRA 21. Now to find out, to make our the electrodes more reproducible, we find we did different experiments, different electrodes, like 8 electrodes we have fabricated. There is some like more their limitation of the biosensor work is that our electrodes their stability, electrode stability, as well as the reproducibility is a problem. If we are making the 50 and the 100 electrodes, they are not giving the same value, same current value. So we have to try to make a similar, bilkul exact similar type of the electrodes. So in our this work we have just tested with only the 8 electrodes we fabricated, and the find out how much the current values it changes. So in this case there is a RST value which is obtained 1.38%. Matlab that is giving these electrodes are good under the means acceptable value. Again, that is a question that how we can say that is a specific binding between the antigen and antibody. So we have to check our electrode this response without antibodies. That is a control experiment. So here you can see in the figure A, the electrode response that is not changing without antibodies onto the electrode surface with the different concentration of the CYPRA21. So that indicate that there is a specific interaction of the antibodies with the antigen that giving the change in the response value. So that give the specificity of electrode. Again, we did some type of the interference. You know that if we are going to for some device, so there are the different type of the molecules, different type of the ions, and the metabolites are present in our the saliva sample. So we did few, not all. So some like biomarkers like CA125 and IL8 also tested. Some ions, calcium, glucose, and NaCl, KCl. We have tested, and you can see that the response only we obtained with the CYPRA21. So that is also giving the specificity of the electrode. Stability is also checked with the up to our 18 weeks. Stability is also main point for the biosensor application. If your electrode is not much stable, that is a no use. So in this case, that we stability is not so much good, but we can be improved if you use the different nanomaterials. So we compare our results, which already published into literature, and our work, which is more sensitive. That is, the sensitivity is around like 12 microampere, and their low detection limit is also 0.001 nanogram per ml. So then second, to improve their sensitivity, again we have used cerium oxide and the RGO based electrodes. And this is a scheme that's showing that how we have fabricated the cerium oxide nanoparticles, and the graphene oxide is in situ, is reduced during the synthesis of cerium oxide. Nanocubes are formed here, and these first we synthesize, and then using the same EPD, here we have used a spin coating thin film prepared onto the ITO surface, and then same antibodies immobilized, and their response was studied using the DPV. Here, what is the importance of the cerium oxide and the RGO? You know that RGO, they are the stuck. If we are using only RGO, the RGO sheets will be the stuck. If we use some nanomaterials, say, in the composite, that these nanoparticles may be inserted into the layers of the RGO, and they inhibit their stacking of the this RGO sheet. So once they are nanoparticles are inserted into the RGO layer, they enhance their electrochemical response. So keeping this point in view, and there is a synergetic effect. RGO is provide the more electrochemical response, and the cerium oxide nanoparticles, they are showing, they are inhibit their stacking of these layers. So both they showing the synergetic effect. So first we have characterized, that is our geo, that showing their peak peak at 266 nanometers, and then we only cerium oxide nanocubes is to like the peak we obtained around 320 nanometer, and the composite also that is showing the both the peak corresponding to the GO and the RGO, that confirm your materials having the both the nanomaterials. Simultaneously, we check the Raman spectra, and the G and D we are present into the GO, and in this case the composite, that G and the D band appeared into composite, and only the cerium oxide nanoparticles peak also are present. So these both the composite formation is confirmed by these techniques. XRD is also showing the peak corresponding to GO and RGO and the cerium oxide nanoparticles. And then you can see here the cubic structure of cerium oxide nanoparticles we are confirmed from the TM. And in this case, you can see in the figure D, that the RGO sheet that is present onto the cerium oxide nanocubes, that is the interface formation take place between the RGO and the cerium oxide cubes. That there is a both their crystallinity of these nanomaterials are visible into the TM study. So XPS also we have did to confirm their molecular binding of these molecules, and there the corresponding peak to cerium, oxygen, carbon is visible in the different spectra. Like you can see here, this is A is a complete survey of materials of the composite, and second is the we have this convoluted this data, and the P corresponding to the cerium that is present in the band gap of the 290 to 280, 284 to 290 in the energy, the binding energy. And in oxygen also showing between the 528 to 536, and in the cerium these ceriums related functional group are present, energy binding are present in the area of the 880 to 920. So these XP studies also confirm there both the molecules are present into this composite. Now this material again we use for the electrochemical study. Then first we did the pH effect, and again in this electrode you can see that showing the maximum current value at the 7.4. So all these material that depends on the materials behavior with the pH. So we have to check there for the pH effect of the electrolytes. So here the 7 point pH is showing the maximum results. Again here we have confirmed from this to how much the antibodies concentration to be immobilized onto electrode surface. You can see that is varies from the 10 microgram per ml to 100 microgram per ml. But after this the 50 microgram per ml is showing the maximum current, and then it start to decrease it. So maximum the 50 microgram per ml this antibodies concentration we have used for making the electrodes. And this on the different electrochemical behavior of the electrodes. Here we have used the cyclic voltammetric techniques, that's showing that how current value is changing at the modification of the electrode. And simultaneously we also can be confirmed using the impedance spectrophotometric techniques. This is the impedance spectra, and you can say that the RCT value, charge transfer resistance, can be monitored. You can see that this RCT value is a cycle. This is a Nyquist plot that's showing that RCT value is varies with the modification of the electrodes. So again, we did that the response study with the a different concentration onto the electrode surface. You can see that their low value is 0.625 uh, picogram per ml to 15 nanogram per ml. So this RGO cerium oxide electrode showing the low concentration of your detection limit, and their wide range, but the higher concentration up to the 15 nanogram per ml we can detect using this electrode. So we just find out how this curve, but here linearity in the two way. The linearity we obtained up to that 0.01, that is showing the linear decrease in current, but after that is from this point again is decreasing. So the linear response into the two way we have obtained, and simultaneously we did we checked this electrode with the this real sample, means the saliva. Students took their own saliva and tested their IL-8, CIPRA 21 concentration into this samples. So that is showing their response. So simultaneously, we did some concentration like the control experiments and the interference and reproducibility also and stability. Now, to make the more the cost-effective electrodes, we have prepared conducting paper using graphene oxide modified electrode. So here, what we did, we took one the paper sheet, that is the ivory sheet, and dipped into the GO, that is the graphene oxide solution, that is exposed by the hydrogen hydrate, and that is showing that from the RGO paper, that is the conducting, that's showing the conducting in nature. This electrode used as the substrate. In the previous paper, we use the ITO. So this paper replaced the ITO. That's showing the good conductivity. So this electrode, this paper-based electrode used for deposition of nanomaterials. Here nanomaterials we prepared. The gold nanoparticles, they are capped with the cerium oxide cerium oxide, no sorry, gold nanoparticles binded with the cipra 20, this L16 as the capping agents. So the sulfur group of the L16 that bind with the gold nanoparticles by the covalent bonding, and they modified their surface and also provide their stability of the gold nanoparticles. So these nanoparticles we deposited onto the this RGO based conducting paper using the electrophoretic deposition. So that confirm that your paper is conducting in nature, and then used for immobilization of the antibodies corresponding to the IL-8. Here we have interleukin 8 biomarkers based antibodies we have immobilized onto this electrode, because IL-8 is also showing the corresponding to the for the corresponding to some disease infection and other inflammatory disease can also give us some signal. So IL-8 biomarker is also very important for the clinical diagnosis. So this is the curve which we have took their response as the concentration is the changing with the respect to current. So now we will discuss the result, that first these gold nanoparticles we have prepared, that we have this peak we obtained at around the 510 nanometer, and after this modification of the gold nanoparticles, their peak is shifted to the 560 nanometer, that indicated their modification of the gold nanoparticles. Then further we during the experiments we also find out their color is the drastic changes after the modification with the L16, and their particle size is distribution, that is the maximum around 2 nanometers we have obtained of the gold nanoparticles, and these are the monodisperse in natures as we prepared. So this paper then we have confirmed with the SEM also using a SEM EDS mapping. So EDS mapping, that's showing the color of your this elements present onto the this surface. So first is the paper, that is RGO, only the RGO paper. Second, RGO paper with the gold nanoparticles with deposited onto that surface. You can see that is a this globular this morphology onto the electrodes, some indicate their nanoparticles are deposited on the electrode surface, and this is EDX showing the carbon, oxygen and the gold present onto the electrode surface, and then this color the photograph showing presence of the carbon, oxygen and the gold onto the conducting paper. So these confirm your nanomaterials your onto the conducting paper, showing all the elements into this. Now that a response study we did, and then again here there is a no any type of the redox species. So that this work also showing without any means mediator free response. There is a no any redox species, and here we have used the chrono amperometric response. You can see that IL-8 concentration is decreasing as sorry, IL concentration is increasing, their current is decreasing. So we have this varies concentration up to the 16 picogram per ml. But this advantages of this work is that this detection range is very less. Yes, yes, sir. Okay, sir. Okay. Huh. So uh, now their detection range is 1 to 9 picogram per ml. That is not sufficient according to the physiological range. And but it's showing a response with the spike sample. We took saliva and IL-8 concentration molecules are added into the saliva samples, but that is showing the response. Then we have again that to make the more useful for means wide the detection range and the sensitivity, we use this silver capped molybdate nanoparticles, and then using the same electrochemical biosensor application based sensor we have developed, and that is showing the very good the detection limit up to the 40 nanogram per ml. That can be used for the further applications in the biosensor. I think time is very less that I want to skip some slides. So second, we simultaneously we are working for vitamin D detection, that is a 25 hydroxy vitamin D3. That is also very important nowadays. I think every person is suffering from the vitamin D deficiency, or sometimes we are not aware, and their testing this cost is also very high. So in this case, we have fabricated electrospinning nanofibers, like electrospinning machine, and then these fibers deposited onto the same the conducting paper we have used, the RGO conducting paper, and then modified with the antibodies which is corresponding to the 25OH vitamin D3, and the chronoamperimetric response was this observed. These are the STM image of the nanofibers, and that we find that these nanofibers showing the 330 nanometer in their diameter. And this is the electrochemical response. The detection range we have obtained 10 to 100 nanogram per ml. Sensitivity is 0.16 microampere. Vitamin D is where only the few papers are reported into the literature, and there is a no any commercialized device is available, no patent. We have filed patent for fabrication of vitamin D3. Here we have used some carbon based cloth, and that is showing the very good sensitivity up to the 2 microampere, and their linear range is also very good, 1 to 160 microgram per ml. You can see the very sharp change in the current with the concentration of the analyte up to the 200 nanogram per ml. Simultaneously, we did the serum sample. Serum sample, we did simultaneously the ELISA, and ELISA results compare with our sensor strips. That is showing the very good agreements. And the last that I want to show some the like carbon more biocompatible carbon quantum dots we have prepared, and the carbon quantum dot base work we are now presently we are working with the Professor Mohanan, and we are collaborating with this carbon quantum dots base work. This is very simple synthesis process. Within the 3 minutes we can prepare carbon quantum dots which are very highly this the fluorescence is nature. Here we have tested their biocompatibility of these this carbon quantum dots. They showing the deuterion, they showing the both positive and the negative charge onto their surface. They easily can be penetrate into the cancer cell lines. So we are synthesized and the confound with the different characterization techniques like the this fluorescence, and they're excited with the different wavelength, and their emission has been this observed, and this is the FTIR. All the corresponding peak is visible into the FTR spectra. This is the very beautiful TM spectra of this carbon dot. Cytotoxicity with the we are done with the two cancer cell line that is corresponding to the oral cancer cell line, that is the CEA8 and the FADU cell lines, and it's showing the very good fluorescence. Generally, people are using different type of the dyes to create the fluorescence, but these are the own itself fluorescence in nature. And you can see that up to that the 400 microgram per ml, it's showing the very good the fluorescence in nature. And time dependent study also did, and up to within the 30 minutes that it start to showing the fluorescence in the both the cell lines. Exocytosis is also checked because nanoparticles nature they are stuck into liver and the kidney cells. So we monitor that is exocyted or not. But they start to exocyted from the cells. From like you can see up in the 48 hours, mostly cells, mostly carbon quantum dots are exocyted from the nature. I think it's enough, and I acknowledge our funding agency, GSC, DBT, USC, and this AIRF GNU have all the facility TM, SCM, and XRD. And these are my collaborator, which I have worked with the Dr. Amit Dinda, Alok Thakkar, Dinesh Mon. Is other work is I have Indo-Russia project with the Professor Sergey from the Moscow. And this is my group at SCNS. The students are working. Sri is working in the cancer cell lines for biosensor. Deepika vitamin D. They are the PhD students. Thank you, sir. Now is open for question.
Beautiful.
Hello.
Hello.
Thank you so much, ma'am, for the inspiring lecture on prospects of nanomaterials in biomedical applications. The interdisciplinary approach converging material properties of nanomaterials for biomedical applications seems quite interesting and throws light on the wide-reaching application scope of the materials. Due to time limit, we are forced to move on to the next session. We are skipping the question answer session now. Sorry, ma'am. And once again, thank you so much for sparing your valuable time
Write
with us.
email to me if anyone want to discuss, and I am ready to collaborate. And if someone are interested, thank you very much, sir.
Thank you, madam. Thank you so much.
This opportunity. Yeah, thank you very much.
I'm handing over the session to Atira. She will be comparing next.
Very good morning to all. Hope you had an informative section by Dr. Pradima. And of course, kindly excuses for the misling happened in between. It is due to some network errors here. So let's move on. And now it's my duty to welcome Dr. Gayatri, the senior scientist, Jawaharlal Nehru Tropical Botanical Garden Research Institute, to invite and introduce our next speaker.
Thank you. I take this privilege to welcome Professor Shyam Sharma. Dr. Shyam Sharma is a professor in Department of Pharmacology and Toxicology at National Institute of Pharmaceutical Education and Research, Naipur, Mohali, Punjab, India since 2009. Before joining Naipur, Mohali, he worked as a postdoctoral fellow at the University of Illinois at Chicago, USA. He did his PhD in pharmacology from All India Institute of Medical Sciences, New Delhi. Dr. Sharma published more than 140 peer-reviewed research papers, book chapters with more than 4,700 citations, H-index 39 and I-10 index 91. His research interest includes understanding the potential role of pharmacological agents in stroke, cardiovascular diseases, and diabetic complications. He has more than 20 years of teaching and research experience. Dr. Sharma has guided more than 100 master's and doctorate students. He has delivered more than 100 invited talks at national and international levels. He has completed more than 20 extramural and industry-funded projects. He is the recipient of Organization for Pharmaceutical Producers of India, acronym as OPPI, Scientist Award, Shakuntala Amir Chand Award, and Dr. D.N. Prasad Memorial Oration Award of Indian Council of Medical Research, CDRI Oration Award, P.P. Surya Kumari Prize, and Professor N.S. Thala Award of Indian Pharmacological Society. He is the Fellow of Indian Pharmacological Society. The topic of presentation for today's seminar from Dr. Shyam Sharma will be cardiovascular safety assessment of new chemical entities. Professor Sharma, Department of Pharmacology, Toxicology, National Institute of Pharmaceutical Education and Research, Mohali, Punjab. On behalf of the organization committee, I welcome Professor Shyam Sharma for the presentation. Over to Professor Shyam Sharma. Thank you.
Welcome, Professor Sharma ji.
Thank you, sir. Actually, first, actually, I would like to thank Dr. Mohan, Dr. Sudha, Dr. Nair for inviting me over here. I'm audible and my slides are visible?
Yes, audible.
Okay. Okay, thank you very much. I think I find the topic of this conference is very interesting because this is the one which is going to cover like food, chemicals, and the nanomaterial. And nowadays, actually, food, nanomaterials are much talked about actually, and that's why I find this topic interesting. When this Dr. Mohanan invited me, and I just looked at it like what I can talk about. So I just took it to like toxicity. When we are talking about the toxicity, I thought like one of the major issue nowadays, everybody or most of the molecules actually they are facing is the cardiac safety of any molecule, whether it's a new chemical entity. And mostly you know that the drug which are discovered initially, they are called new chemical entities. And later on, like when they have gone through preclinical, clinical studies, then only they become drug when they are considered to be the efficacious and the safe. This is the drug discovery and development pathways. I think most of you would be knowing. I'm not going to into the detail, but I would like to mention that these safety studies need to be performed, like preclinical safety study can be performed before submission of IND, and then you also need to perform some of the follow-up study during the clinical phase also. They like these safety studies are required to be followed because like during clinical trial, if there's a cause of concern, then again you have to conduct these clinical or preclinical studies over there. And at the end, like you can see, like about 10 to 12 years it takes to file the NDA, new drug application. So you can look at it like IND, investigational new drug application, and NDA, new drug applications that need to find, and that's involve all preclinical as well as the clinical studies. One of the main reason of attrition, you have seen that a lot of drugs actually which reach to the clinical trial, but they fail to show up in the market. Why? Because there are a lot of issues actually which are associated with this. I can tell you one of the disease condition like stroke. Till today, like we do not have a treatment for stroke. Why? Because most of the drug like which have developed in the preclinical, and when they are reaching clinical trial, they have been withdrawn from the market because none of them is found to be efficacious in the clinical setting. So there are several of these reasons which have been shown over in this slide. Attrition in drug development. You can see like most of the cases, you see the clinical efficacy, 22.5% of the molecules actually are withdrawn due to clinical efficacy. Portfolio consideration, sometimes like me too drugs are there already, the better drug has come up in the market. Clinical safety, toxicology, and various other like clinical pharmacokinetic, bioavailability, efficacy in preclinical studies is not good. Okay, formulations, you are not able to make the desired formulations, and some regulatory and the patent issue that some of these things are not able to come. And one of the historical perspective about this these studies, what I'm going to talk today, that serious injury or the death of the volunteer and the patient participating in clinical trials are very disturbing when it's occurred. The organ system and the functions most frequently responsible in these events are cardiovascular, CNS, and respiratory and renal. There are like three major systems like cardiovascular, CNS, and respiratory, which are being considered to be the vital organ of the body, and any alteration in their functioning can lead to problem like cardiovascular. There are several of these drugs have been withdrawn from the market because of cardiovascular QT prolongation. And most of the time, this tox study, I think before me, Dr. Vikas Meriwar talked about some of those tox studies where like acute, subacute, and chronic toxicity need to be done. And those studies do not look at the functions like whether these molecules are going to alter the functions or physiological functions of the body. Now you can see like cardiovascular safety is one of the major concern over here, that what we have seen that uh, most of the drugs which have been withdrawn from the market, you just name few of the drugs like blockbuster drug, like most of you would be knowing about the terfenadine. Terfenadine was anti-allergic drug actually, which came to the market very effective, but later on it came to the knowledge that there is a QT prolongation and death of some of the uh, uh, healthy volunteer or healthy patients have occurred during the clinical phase or post-marketing surveillance. So that was the major issue actually, and that is why terfenadine was withdrawn from the market. And now in the market, we have like terfenadine metabolite like fexofenadine, which is being used, or otherwise people are using other anti-allergic like cetirizine or levocetirizine. So now, if you look at the drug which have been withdrawn from the market, like prenylhamine, liroflagin, tramadine, cetindole, terfenadine, estimizole, another like anti-allergic, antibiotic, prokinetic agent like cisapride, droperidol, and levoastrolol. Now you can see like these molecules, uh, the drug discovery costs like about $1 billion, which is projected, or more than $1 billion. And you see like if these drugs are reaching clinical trial and if they are withdrawn, there's so much of the loss of the money to the company. Even like there was a concern like cause related with the COVID-19 when this hydroxychloroquine and azithromycin combination was used. Like there was a paper which was put in the Nature Medicine in 2020 after this COVID-19 emergence or pandemic. The QT prolongation or QT interval patients with COVID-19 treated with hydroxychloroquine and azithromycin. Ventricular arrhythmia risk due to hydroxychloroquine and azithromycin treatment. And you see the list of the drug actually which have been investigated in time of this pandemic. What we have to always look into it, like their safety and the efficacy need to be planned, so that there should not be any undesirable pharmacodynamic effect, or there are no undesirable QT prolongations actually in the patients. So that's why you can see like cardiac injury and myocardial suppression with chloroquine, hydroxychloroquine, remdesivir question mark, because some of the investigation still need to be done. Prolonged QT prolongation is there with chloroquine, hydroxychloroquine, favipiravir, lopinavir, azithromycin, moxifloxacin. So that way, these are the one actually which indicate like there is a potential cardiac adverse effect can be there. So that is why, like before administration to the patients, actually, which are already already on several other drugs, and you are putting these drugs actually that can cause QT prolongation. You have to think a lot before administration of this, their history, or their cardiac test, and all everything need to be looked into. To look into this issue of like QT prolongation, ICH International Conference on Harmonization, now is International Conference Council on Harmonization, they came up with harmonizing the different guidelines. Actually. So this is the harmonization by ICH. You can go to the websites of ICH, www.ich.org. And in that, you can find a lot of information about what are the studies which are required for safety and the efficacy of any molecule. All the guidelines are available for conducting those studies. Those who are interested can go through these guidelines. I'll talk about like the regulatory guidance which came in the through the ICH S7A and S7B. Safety S7A guideline and S7B safety guideline S7B. So S7A guidelines basically the safety pharmacology studies for human pharmaceuticals, and particularly for QT prolongation, because that's a major issue and there was a death was reported in the some of the patients. They have brought out a separate guideline like non-clinical evaluation of the potential for delayed ventricular repolarization by human pharmaceuticals. And even like for clinical studies, there is ICH E14 guidelines. So now the ICH guidelines are like harmonized guidelines. They are being followed throughout the world. And most of the time, like your Japan, US, UK, Europe, all of them are following these guidelines. These are E14 guidelines for clinical evaluation of QTC, interval prolongation, and pro-arrhythmic potential for non-antiarrhythmic drugs. These are the objectives of these guidelines, like they investigate undesirable pharmacodynamic effect of any new chemical entity, evaluate adverse pharmacodynamic effect, investigate the mechanism of the adverse pharmacodynamic effect, and major objective is to protect clinical trial patients from potential adverse effects of pharmaceuticals. Okay. So now, like, if suppose somebody asks you, okay, like, just can you involve in clinical trial? Only like if you see like those people, those who are educated, they will not at all. They will see, okay, no, I will not involve. If they have to be involved, then what? Then there's a consent form. Without consent, actually, you cannot conduct those studies. And that is why these guidelines are required actually before administration of first-time administration of any drug. Like phase 1 clinical trial, you require all these studies. Before phase 1 clinical trial approval, you require these studies. So these guidelines is basically like prescribed for new chemical entities, biotechnology derived products for human use, and even the like since this was the conference related with the nano also. The nano formulations also required to follow these guidelines, because when you put in nano formulation, there is a change in the dynamic, they change in the efficacy, there may be change in the toxicity. So that's why like you require even for the nano formulation. If you are changing any formulation, you require these guidelines. So these are the consideration which have been put in the test system, what you require, like which animal you require, which species you require, some of the species which are not sensitive for that, what kind of gender, age, test system susceptibility, sensitivity, reproducibility you require. So what is the system you have to use there that you have to think before the study. And you can use range of in vitro and in vivo studies for this. And dose level of the concentration of the test substance, what you require. For that, actually, you would have already conducted some of the dose response studies for efficacy studies. From that, you have to draw the dose for in vivo studies. Duration of the studies on metabolite, isomers, and finished product. If you are changing the bioavailability of any formulation and of any finished product, you need to do the safety pharmacological study. And the condition under which the safety pharmacological studies are not required. For locally applied agents, you don't require actually this one. For cytotoxic agent for treatment of end-stage cancer patients, and for cytotoxic agent mechanism of action, there may be the value in conducting pharmacological. New salt having similar pharmacokinetic and pharmacodynamic effect. That's also exempted from that. What is the time actually like of conducting these studies? Lead identification, you can do the HERG cell line based studies, and then you can do the patch clamp study during lead optimization of these new chemical entities. Small animal cardiovascular studies can be done during that time, and even in-depth cardiovascular profiling need to be done in non-rodent like dog. And then preclinical development, you require the GLP as per the regulatory guidelines. All like whether Indian regulatory or maybe other country regulatory, most of those regulatory guidelines require the GLP for cardiovascular safety pharmacology. So there's the timing. What is the timing like these studies need to perform? The timing is before first time administration to the human, any new molecule. Okay, before administration, that you need to do. And even during clinical development, if there is a cause of concern and suddenly like one death occurred due to some cardiac failure or QT prolongation, then again you have to go back and you have to look for what could have been the mechanism why there was a failure to detect in that particular state. This is the hierarchy of safety pharmacological studies. You can see the core battery studies, follow-up studies, and supplemental safety. Core battery studies basically involve three major organ systems. Okay, they are the one actually vital organ system like CNS, brain, cardiovascular system, and respiratory system. Okay, and these core battery studies need to be carried out under the GLP format. Follow-up studies, if there is any cause of concern in the core battery study, then you need to do follow-up studies like CNS, cardiovascular, and SPD, follow-up studies for the in-depth studies that you can do. And some of the organ systems which were not covered because these were only the vital organ systems, supplemental safety pharmacology like GI toxicity, ANS, renal, urinary, and other like immune system that can be done in supplemental safety pharmacology studies. Like suppose celecoxib is causing GI damage, you can do safety pharmacology in cases of supplemental studies. These are like cardiovascular safety pharmacology, core battery, and the follow-up studies. What involve core battery study? You need to investigate the new chemical entity, whether they have any effect on the blood pressure, they have any effect on the heart rate, they have any effect on the ECG. And in follow-up studies, then you can do if there is a cause of concern, there is a change in the blood pressure, heart rate, and ECG, you can do cardiac output studies, vascular resistance, ventricular contractility, and the effect of endogenous or exogenous agents, actually whether they alter new chemical entity-induced effect. That way, like adrenaline, noradrenaline, or sodium nitroprusside, like nitric oxide, whether they are changing, you can use them for these kind of studies. So they like cardiovascular safety pharmacology. As I mentioned, like since there was a major concern about the death due to cardiac arrhythmia in the clinical trial and the post-marketing surveillance, that generally we call phase 4 clinical trial. But during phase 4 clinical trial, terfenadine was launched in the market, everybody was started consuming it, and at the end, actually, like what happened, some of the death were reported. And later on, again, regulatory guidelines, regulatory agencies got active and they have to decide to go ahead. So for that, like so that there's no withdrawal from the market, actually, like they have considered these guidelines so that at the before you starting the clinical trial phase 1, you need to conduct these kind of studies so that humans are safe during the clinical trial. And these guidelines like non-clinical evaluation potential for delayed ventricular reproduction, QT interval prolongation by human pharmaceutical. This applies to new chemical entity and marketed pharmaceuticals, and major objective is to identify the potential of the test substance to prolong QT and to relate the extent of QT prolongation and the concentration. This is like a strategy which is being used. Like here, we are going to use all sort of like in vitro and the in vivo studies actually, which can be used for determination of whether any molecule is going to cause QT prolongation. And these guidelines, you can, if you go to the ICH website, you can find these guidelines and you can definitely read in detail about these guidelines. The what sort of like in vitro and the in vivo studies which are being conducted under this S7B. In vitro, like you can use heterologous expression system like CHO cell line can be used, HEK cell line can be used, and these cell lines has to be transfected with the one of the channel like HERG, human ether agogo related gene, and this is the one actually like responsible for the potassium channel. If any drug is going to block potassium channel, that can lead to prolongation. That's the one thing which have been come out from the research. Cardiomyocytes can be used, Purkinje fibers, papillary muscles, perfused myocardium, intact heart can be used, and in vivo electrophysiological studies, QT prolongation, ECG in animal models can be done there. When we are talking about this in vitro hug assay, like patch clamp, if you look at our country, I think like very few labs, maybe not more than 20 labs throughout India would be having this patch clamp facility. And most of them, they are involved in the ion channel research. If you are going to work on sodium channel, potassium channel, calcium channels, you require patch clamp setup. And there are now the manual patch clamp and there's an automatic patch clamp setups which are available. One of the patch clamp setup, we also have this manual patch clamp, and we are using for several stages. So these patch clamp are basically the gold standard which can determine whether there is a blockade of the potassium channel. Or there that you can see, like all of you, I think, since researcher, you must be knowing about like patching a single cell and do the ion channel studies. But like these techniques have a low throughput. Like when these companies come, like if you have like hundred of molecule and thousands of molecule, you require a patch express or kind of a automated system where in a day you can do many molecule patch clamp studies. So that way, like most of the time, companies like Pfizer and all, like they are using these kind of studies. High throughput QT screens are there for millions of molecules. Most of the companies are synthesizing throughout the year. They would be having these kind of. They don't conduct for each and every molecule. The molecule only which show the which is potent and show the best efficacy on those molecules. They do QT screen. And if that molecule is showing QT prolongation in this particular screen, they will drop that molecule. Whatever the case, actually, they are going to drop the molecule because these company can't bear the loss of like $1 billion actually at the end of so when it comes to validation, several of the molecules have been used, like this kind of a investigation you do with the concentration and different molecules. So now you can see like this is one of the company like MDS Pharma Service they provide. So now even like you can set up your own HERG assay lab, and most of the Indian company can come to you for, or maybe international company they can come to you for this kind of assay, because this is a regulatory requirement. Regulatory requirement is there for this. Estimajol, cisapride, E4031, it is again one of the antiarrhythmic. Haloperidol, ketoconazole, pimozide, quinidine, risperidone, pirfenidine, verapamil, moxifloxacin. So if you look into the patch express assay and the conventional patch clamp assay and the radioligand binding assay also can be performed, you can find estimajol, cisapride, E4031, some extent haloperidol, terfenadine, they are causing QT prolongation. But ketoconazole, moxifloxacin as such do not cause. Ketoconazole as such do not cause. But when you combine it with terfenadine, it can cause QT prolongation. So there, like ex vivo studies can also be done on isolated rat heart, manual Langerhoff preparations, screen neat and automated computerized model which can be used for investigation. Then investigation of the action potential duration during isolated retrogradely perfused intact heart can be done, and that can also be used for investigating. These systems are devoid of metabolic, humoral, and nervous system because like these are isolated system, and that is why like when we think about felines and this, we always prefer like can we use some animals for these. For QT prolongation of ECG, whole animals, you can use beagle dogs. These are like kind of a docile dog, and you can work on them. Cynomolgus monkey, rabbit, guinea pig. Rat are not suitable animal model for doing QT prolongation. That's why like you have to always look into whether which species is going to be better. You can use conscious or anesthetized or the radio telemetry study. That way, like in our facility, we use generally guinea pig for doing these kind of QT prolongation studies, as we do not have a big dog. But like some of the company like Zydus Cadila, they have a dog facility and they do the rat telemetry studies, where like you can see this telemeter, this kind of telemeter is transplanted in the animal body, and those if you see these kind of like cannulas, they are connected with their arteries, like for recording of blood pressure, attached with the cardiovascular system for recording ECG. Continuous recording can be. I think most of you remember this kind of a like ECG wave, like P, Q, R, S, T. So this is the QT where you can see like QT is the one. This particular like duration is generally, or QT prolongation. This is generally increased in case of these. If with any new chemical entity or any drug, then there is a lot of consideration need to be considered, like at what level, so that like need to be decision need to be taken to withdraw the from market. That need to be. The ventricular action potential, you can see like there's a slow repolarization actually with this red line you can see. Hard current normal, this is the blue, but like LQT is like you can see QT prolongations actually this red one, this is delayed. And then also you can see the prolonged QT in the C, where you can find there's an increase in the QT, okay, and normal rhythm and torsade depointed phenomena. And because of that, like a lot of cardiac arrhythmia and the death has been reported. Some people nowadays trying whether zebrafish can be used, but again, like the regulatory agencies still have not considered this method. They are just under evaluations and you can't use. Okay. In case of like heterologous expression system, you can use some of them actually. Human stem cell derived cells can also be used. Purkinje fibers, papillary muscles, perfused myocardium, intact heart can be used. Even many like computer modeling people or computer experts actually like software engineer, they are developing like in silico method. Like, can we assess this toxicity by just putting the structure of the molecule in the computer? But again, like at the last is a computer can do amazing thing, but even the most powerful computer can't replace animal experimentation in the pharmaceutical research till date. This is the one. There are like some of the equipments which are being used, like this is a non-invasive blood pressure recording instrument in which like we record the blood pressure using the tail vein, like tail artery. So like tail is put on this one actually, and then you have to record the from the tail artery the blood pressure changes. This is called non-invasive blood pressure monitor. Is there invasive blood pressure monitor also there? Like we here, like we need to perform the surgery, and then we isolate these common carotid artery, and we cannulate the common carotid artery, and from common carotid artery we record the blood pressure changes. And ECG changes, you can put the like electrode outside. There's no need to perform any surgery, but you can just anesthetize the animal and put the electrode and do this. Patch clamp studies can be done for doing the Hurg assay. And then these are kind of telemetry devices like one of the company like DSI can provide actually, and they're kind of a transmitter. These are different kind of transmitters which are showing. These, you have to perform a surgery in dog, and then you have to implant these transmitters inside their body, and then you are going to record this. What is the main thing actually which is I would like to mention about that these studies, if you have to submit these molecules for regulatory approval, like for phase 1 clinical trial, then you require these core battery safety pharmacology done under the GLP in the GLP certified lab. And I can proudly say, like Dr. Mohanan has a rich experience, I think more than 20 years experience as a GLP inspector. So now you can see like Sri Chitra having so much of like expertise in GLP. Being a GLP inspector, he has conducted a lot of inspections, and I can say that he's the pioneer in GLP studies. And word like GLP is a quality system concerned with the organizational process and the condition under which non-clinical health and environmental safety pharmacology studies are planned, performed, monitored, recorded, archived, and reported. And I think like every one of you, like since it was mentioned, the chemical, like all like pesticide product, pharmaceutical product, cosmetics, veterinary drugs, food additives, feed additives, and industrial chemicals, like if they have to do like non-clinical safety studies, they need to definitely look at to conduct these studies under GLP. In India, like there's a National GLP Compliance Monitoring Authority that take care of giving the GLP certification to the lab. And I think by this time, more than I think 30 or 40 labs are there in India which are GLP certified. And I can say NIPER was the first government laboratory in India which was certified for the GLP for conducting acute, subacute, and chronic toxicity studies. So this indicates that in India also have a lot of strength actually for conducting these studies. And that is why like OECD principles of GLP, I think like everyone, whether you need to do the GLP study or whether you are doing regular your master's or PhD research, you must read these OECD principles of GLP because they are going to be very good for every researcher, what I feel like. Because that increase the probability of reproducibility of your studies, and you are going to maintain the data, all the data for 10 years, 20 years, because you can see nowadays a lot of distractions are there, and they ask the data, okay, where is the data and all, and so many people will say, oh, we don't have data, it's already more than 10 years. How? But like GLP study makes sure that you maintain all the documentation. And that is why sometimes we call GLP, give lots of paper, because a lot of paper need to be generated and all. And these principles actually basically involve in all facets, whether on facilities, on the organization, records and report, equipment. You have to create standard operating procedures, test system like your rat, mice, dog, or anything, or test and reference items. Like you have to have the GLP. I think every one of you must go through if you're not gone through. Some of the study, like I would like to mention about cardiovascular safety pharmacology and the curcumin nanoformulation we have done some time back. How this need to be standardized? Like I'll tell you, like how you have to standardize the cardiovascular safety pharmacology. Now, in this particular study, we have used phenylephrine and sodium nitroprusside. If you know that phenylephrine basically involved as a vasoconstrictor agent, and sodium nitroprusside basically involved as a vasodilator. So this red line is the normal animal systolic blood pressure in the red, and phenylephrine is 1, 3, and 10 mg per kg. You can see the green line, there is an increase in the pressure, is there, systolic. And the black line, there is a decrease in the systolic pressure by sodium nitroprusside, which is because like that's release nitric oxide, and nitric oxide is one of the vasodilator. So this is like upper one was systolic and down one was diastolic pressure. Again, you can find like similar kind of effect on these kind of changes. Then mean arterial blood pressure, and this is generally like calculated on the basis of like your diastolic pressure plus 1/3 of pulse pressure. Pulse pressure is the difference between systolic and the diastolic pressure. And then there's a heart rate. Now you can see these animals most of the time a heart rate of 350 to 450. Okay, and whether like phenylephrine and the sodium nitroprusside, like the effect that you have to conduct. What like in this study, we have used various formulation, nanocrystalline, solid dispersion formulation. These formulations were prepared by one of the expert in our institute, Dr. Arvind Bansal. He is the expert in pharmaceutical formulation development, and their labs actually synthesized these formulation. They made this celecoxib formulation and as well as the curcumin formulation for investigation of the safety pharmacology. That we conduct all those safety. In this like celecoxib formulation given at 20, 40, and 120 mg per kg, and we did not find actually much effect on blood pressure and the heart rate. Now you can see on the blood pressure, actually like mean arterial blood pressure and the heart rate, we did not find. And that's why like we have to always certain that. But when we have checked like their GI safety, okay, gastric emptying and all those, there like we find some effect, not here. And again on QT prolongation, like where like you can see the QT prolongation also was not observed, actually not no change was observed with the QT interval actually in this studies. Curcumin, which I mentioned that curcumin formulation like nanocrystalline solid dispersion formulation was assessed for blood pressure and the heart rate, like systolic blood pressure and the diastolic blood pressure. And you can see like various doses of these 50, 100, and 200 mg per kg were investigated, and we did not observe effect. Mean arterial pressure, you can see again 50, 100, and 200. And you can see different time interval. After the administration, we have checked actually up to what time actually like that. So we observed for 2 hours, there were no change. Then on QT prolongation, the heart rate, up to 120 hours, we did not find any changes. If there are any change we observe, actually, then we tell them, then they have to change the formulation. Okay, this is the component is causing. Now, they also come up with another formulation where they curcumin with piperine, nanocrystalline solid dispersive formulation. That's also was investigated for blood pressure and heart rate. And here, we did not get any effect on all these parameters. And that is true, you have to do it actually for this. But if you use some of the drug like terfenadine and ketoconazole, you will get prolongation. So then, I can tell you, initially when we set up our laboratory, we have set up some of the data of existing drug like terfenadine and ketoconazole, whether there is any QT prolongations observed with this one. So there, we use the telemetry device for this study in the animals actually, and then we investigated these kind of studies. If you look at it like ECG morphology in guinea pig, the C panel, you can see. C panel is the one actually where we have used the combination of ketoconazole and terfenadine. And you can see very clearly there was a QT prolongation. A is the terfenadine alone, B is the ketoconazole, and the C is the ketoconazole, terfenadine, and D is the normal. So now you can see if you compare with the D, and you can find there's a QT prolongation observed with this combination. These kind of studies and this kind of a quantification of QT prolongations that has been tabulated over here actually, and this research actually we published. So now they're changing, they're a lot of technological changes are happening throughout the world. Now you see artificial intelligence have come up. People are using all sort of investigations of this technology. In vitro automated high throughput patch clamp already being developed, and they are modifying it. Can we do more number of compounds? External telemetry with high definition oscillometry. So previously, we have inserted the transmitter in the body of animal. Okay, after every 3 months, we have to take out, we have to do the surgery, and we have to change the battery. But now I think technology have come up actually from outside only. No need to perform surgery. From outside only, you can charge those batteries, charge those devices, and there's no need to perform. In silico computer modeling, long way, many companies like TCS and all, they are working to find out can we have modeling. And then again, stem cells, human embryonic stem cells, cardiomyocytes, they are also being investigated for this one. So that's with these, I would like to summarize. All the new chemical entity and the nano formulations need to be investigated for undesirable pharmacology. Safety pharmacology, I see it, S7A and S7B guidelines evaluate pharmaceuticals for cardiovascular safety. Even I can tell you even the food nutraceuticals, some of the nutraceuticals study or some of the Ayurvedic or herbal formulation studies, they are also conducting these kind of studies to make that their formulation is safe for administration. So that's give the confidence in the consumers or buyers actually, this has been tested there no cardiovascular health risk from this formulation. So that's why these guidelines can be incorporated in it's not only for nano formulation or new chemical entity, even for nutraceutical you will find on the internet a lot of companies they are doing this. Safety pharmacology is relatively young discipline in a rapid phase of growth. So there is a safety pharmacology society. USA is there. They are taking a lead. In India, we have a DBAT, diplomat of American Board of Toxicology. Similarly, in India also we need to start kind of a similarly. Major role of safety pharmacological studies in continued implementation of ICH-S7 guidelines and introduction of newer technology offering and improved ability to study the compound. Can we have nowadays a lot of stress always there on animal, can we reduce? Future of safety pharmacology will not only be based on the needs that are defined by the contribution to drug development, but also through the creative and the explorative directions defined by the university. Safety pharmacologists has a great responsibility to protect the public and where the caution is appropriate based on a potential adverse effect. Like they should not hide, they should, whatever the visuals are there, they need to show, okay, if it is causing QT prolongation, then respective measures need to be withdrawn, or maybe some other kind of a molecule need to be developed for these kind of studies. With this, I would like to thank my research group. Actually, one of these celecoxib and curcumin study was done by Richa. I thank her actually like for this kind of study, and all students are students. Thank you very much. And this is not as beautiful as Kerala, because Kerala is considered to be the paradise. Okay, but on campus, Naipur campus is also quite green actually. With this, I would like to thank you. And if you have any questions, actually, I would be happy to answer. Thank you very much.
Thank you.
Thank you, Dr. Shyam, for volunteering your time to share such an informative lecture on cardiovascular safety assessment of new chemical entities. So the presentation gave us an in-depth idea of the importance of cardiovascular safety assessment of new chemical entities, highlighting the current gap in the drug safety. So I believe that there are no queries from the audience. So let's move on to the next section. So since there are no questions, now I'm handovering the section to Miss Ashtami.
So Professor Shyamji, thank you so much.
Thank you, sir. Thank you, sir, giving me the opportunity. Thank you.
We'll see you soon.
See you, sir.
Okay, thank you so much.
Namaste. Now, to share a few words about the final speaker of this webinar, we invite Dr. Jose Priya, School of Biological Sciences, Central University of Kerala. Over to you, ma'am.
Just pray, you can start. Yes, sir. You can start.
Professor Pratap Kumar Shetty is currently the Dean of Research at Pondicherry Central University. He is a professor there at the Department of Food Science and Technology. Professor Shetty has 23 years of research experience.
It's not audible clearly. It's not audible. You can start again.
Professor Pratap Kumar Shetty is currently the Dean of Research at Pondicherry Central University, and he is a professor there at the Department of Food Science and Technology. Professor Shetty has 23 years of experience in research and 19 years of postgraduate teaching in the areas of food science, food technology, and food biotechnology. He received his PhD from the National Institute of Nutrition, Hyderabad, and he started his career as lecturer at the Department of Microbiology at Mangalore University in the year 1997. He pursued his research at the University of Texas Health Center, USA, as NIH Postdoctoral Research Associate. In the year 2004, he moved to Denmark and worked as assistant professor at the Royal Veterinary and Agricultural University at Copenhagen, and then moved to Aarhus University as senior scientist. Professor Shetty successfully guided 10 PhD scholars, 65 master's project students, and he also mentored 6 postdoctoral scientists to date. He has published more than 60 high-impact publications, applied for 2 patents, edited a book, written 5 book chapters, and delivered many invited talks and lectures. A sensor developed by him for the detection of artificial ripening of fruits is being commercialized and being co-developed with an industry. Professor Shetty has been awarded the Fellow of Royal Society of Chemistry UK, and he is very active in professional bodies such as Association of Food Scientists and Technologists and Nutrition Society of India. Professor Shetty is a science communicator. He widely traveled nationally and internationally, covering more than 15 countries, and he is fluent in 6 languages, including Danish. His topic of presentation today is food risk assessment. And now, on behalf of the organizing committee, I welcome Professor Shetty for the presentation. Over to you, sir.
Thank you.
Yeah, Professor Shetty, welcome.
Yeah, good afternoon, Professor Mohanan. And at the outset, I would like to thank Professor Mohanan, Professor Nair, and all other colleagues for giving me an opportunity to share my ideas on food risk assessment to you all. And can I, okay, now it is.
Yeah, you can share your slides.
Yeah. Okay. Are my slides visible? Can you see my slides? Dr. Mohanan?
Yes, it's visible. Visible and audible.
Very good, very nice.
Thank you.
Yeah, and being the last speaker, I think I will be, hopefully I will be able to stick to your timeline of 40 minutes and try to wind up, well in time, similar to what Dr. Shyam has just done. Anyway, I have seen the list of speakers who gave talk on various aspects, especially on the drug as well as nano toxicities, and also there was one on food contaminant also. And in today's talk, I will be talking pretty in general about how a risk assessment is done with respect to food. And if you are seeing here with respect to food safety, the overall utmost interest is securing or saving the consumers from getting any kind of foodborne illnesses. So either something very minor, starting from vomiting and gastric disturbances, all the way to death, which can happen from consumption of contaminated or poisoned food. Even though we are talking about food safety in a food which is consumed, sold commercially and consumed, but food poisoning has been very well known from prehistoric days, where intentionally people used to politically assassinate somebody by putting something into food. Therefore, we are not talking about that aspect. We are not talking about criminal aspect of safety of food. We are basically talking about accidental or natural contamination of food, or unintentional contamination of food, and to some extent intentional, where the food adulteration comes into picture. If you are talking about the concern about the safety of foods, it is not just limited to developed countries, but it's a worldwide affair. It's something to everyday's life. It is not something to do with somebody who is rich or somebody who is poor, but it is about everybody. Therefore, traditionally, if you see, food has always been cooked at home and consumed. And as we evolved into a society slowly. Anyway, it has been many centuries also since we started processing the food and and preserving it for a long time at home. Then started processing and preserving in a place where you can go and buy it. And now, for the last several decades, we have been by and large, taking the food, buying the foods which are processed remotely, where we might know where it is being processed, or in many cases we may not even know where it is being processed. This particular picture, if you see, is a very interesting one, where you will be seeing, you are seeing a mother giving a food to the child. And the child immediately opens her mouth, unless many times, yes, she may not open the mouth if she don't like the food, but otherwise the child just eats the food what mother gives to her. This is what we call the 100% trust. Child will never distrust the mother when the mother gives the food. She may not like the food, that's a different thing. Therefore, in food safety, the most important aspect always is the trust. Therefore, many times you may be knowing, you might go and buy food products which don't have FSSAI marking. However, you will go and buy it still because you are buying it from a source which is a trusted source. In fact, when we were in Denmark, there was one company called Gammeldansk. They used to produce jams and jellies and so on and so forth. But theirs is one of the most prized product only because they have been there for 300 years in business. What does that mean? If a business has existed for 300 years and has a good name, only means that they have gained the trust of the people. Therefore, this trust actually comes from, traditionally from the quality control, how they are able to control the quality, how they are able to maintain the quality. But as the society became more and more modernized, food safety regulation became more and more modernized. Actually, we have now moved to an era of quality assurance or food safety management from a food safety quality control system. And this is also another example which I like the most, because when somebody gives you 100 rupees or 500 rupees note, there is something inscribed on that. I promise to pay the bearer some of this much amount of the money. That means even though it's a piece of paper, but it is the assurance given by Reserve Bank of India. If it is 500, it is 500, nothing less, nothing more. That means this is what exactly is the trust we are talking about. Traditionally, when the food safety has been managed by regulators, it used to be always a reactive approach where the responsibility is with the government. That means food inspectors are supposed to go inspect and say that product which is sold in this particular market is not good, and either the market itself is punished or the person who has made it is punished. Therefore, there used to be no structured risk analysis there. Therefore, here, the entire system used to rely on the end product inspection and testing rather than the process itself. Therefore, as most of the developed societies and developing societies moved on to the modern food safety system or the food safety and quality assurance rather than the reactive approach, here it is a preventive approach where the responsibility is called a shared responsibility, which addresses the entire chain from farm to fork or farm to the table. That means starting from production all the way to the consumption, every step is regulated. Every point is considered as a control point. For example, even the person who, today, it is not just the person who processes the food, but everybody in the chain, starting from the person who is storing the product, who is transporting the product, who is keeping it in the shelf, who is giving it to you, everybody has to be covered under the Food Safety Act. That means here it is entire chain which is looked into, and it is called as shared responsibility with respect to food safety. Therefore, this is actually one more important highlight here is that this is totally science-based, where there is a use of structured risk analysis. It is not just few administrators sitting and saying, okay, this is approved, this is not approved, but it's totally science-based. Therefore, highest amount of scientific trust is given to the assessment of risk from food origin. This also establishes priorities for integrated food control, which relies on the process control. That means you are not traditionally in a food industry, there used to be something called quality control department. And it was the responsibility of quality control department to say that my product is safe. And as far as the regulator is concerned, it is a food inspector. You know how it was done traditionally. But the whole thing has changed today, where there is something called process control. That means it is not just the quality control of the industry which is responsible, even from purchasing of the goods all the way to processing and comes out till it comes to the shelf. Their response, once it comes to the shelf, responsibility of storage shifts to the supermarket. Therefore, when you are talking about the modern food regulation, we call it as the food chain approach. Food chain approach means starting from the production all the way to consumption, that is transport and processing, retail and storage, and preparation, and all the way to consumption. That means every stage is considered as a control point, out of which are those critical control points which will be already identified based on the product, based on the process. Therefore, it is always assumed that food safety hazards may arise at any stage of the food supply. Therefore, both at the public and private level, this food chain approach is applied. Therefore, a preventive approach based on risk analysis rather than a reactive approach. It's not that after something happens, I go and look back. And traditionally, even after something happens, there is no way you can go and look back. Therefore, there was a bottleneck called lack of traceability, which is also addressed today. In a modern risk assessment system, highest importance is given to traceability. You can even trace the product by batch number all the way to the industry. You can go and try to find out based on the record kept in the industry why this particular batch has a problem. In fact, in some of the industries, for example, I will take the example of Jain Irrigation, who are the leaders in mango pulp industry. They have developed a system called Global GAP, Global Good Agricultural Practice, which has been linked to their processing. Therefore, in case of any problem, they will be actually able to track all the way to the tree from which the fruit has been picked. Therefore, that is called as the traceability. And we all know about this dominoes effect. Therefore, at any time point, if anything is wrong, it gets carried all the way to the end product. Therefore, it's actually a rather an unfortunate situation because of various political compulsions. As far as India is concerned, our food safety regulation will not start from the farm, but it starts from the post-harvest, because food safety regulation do not apply to farm, because you know that when we are talking about a farm and farmer, it is more complicated subject. Therefore, there was, with the Green Revolution coming up, there was supposed to be a system called Good Agricultural Practice, which was supposed to be implemented by the Indian farm sector, which was not done. Therefore, in case of our Indian agricultural producers, even though we are the largest producers of many primary agricultural producers, many of those things cannot be used, underline, cannot be used as an ingredient for export-oriented industry. For example, we are largest producer of milk. Therefore, it is very difficult for us to export a product which is milk-based, in spite of us being one of the largest producers of the milk. In fact, you might be quite surprised to see that many of those, Indian milk-based products, when they are being exported, they actually use imported ingredients. That is because, anyway, it will be totally a different topic to talk about. But what I'm trying to say, there are a lot of bottlenecks in these that what did not get controlled at the farm level carried away all the way to the processing, and also which gets linked to the export stage. Therefore, when we are trying to regulate the food safety, there are three approaches in achieving by any of the developing and developed nation. That is, one is reward, educate, and punish. Because traditionally there used to be the punishment. That is, okay, you go and randomly catch the food products and punish the person. In fact, there was one interesting case from Kerala itself. When we study food safety, we use it as a case study. Somebody was penalized for using excess salt in a product. That case ran for more than 20-25 years. Therefore, today the whole thing has changed. As per the modern food safety regulation, number one is reward. Next is education, because in fact, 2/3 of the importance in the modern Food Safety Act is supposed to be education of the consumers and also education of food business operators. That means food business operators means the person who is involved in any component of the food processing chain. Therefore, education, reward, and punish. In case whenever the mistake or whenever the act is found to be illegal or intentional or illegal, then it will be punishment also is used. And unlike earlier, there are specific punishment based on specific violations, specific punishments based on the people who are going to be expected to be affected. For example, as per the Food Safety Authority, the persons who are food business operators either have to get a registration, state license or central license based on how much is their turnover annually or the number of people they are reaching. therefore, or the volume of the business. Therefore, smaller the business, smaller will be the outfall of a unfortunate incident. Larger the business, larger will be the follow-up of an incident. For example, you are all aware of the classical case of Maggie and Nestle, which happened just a very, very recent memory of ours. All of us know very well how much of the issues which were created, either because, which may be an error also, and also legal confusions which was created on the way. Therefore, there are a lot of driving forces in the risk management. Politics, which is the most important one anyway. Press, in fact, press has been one of the most important component today in the food safety regulation, from both the point of view of bringing forward the lacunae and problems, and also creating awareness, and also on the wrong side they also create a lot of wrong notions and confusions in the people based on half knowledge or half cooked information. Therefore, people are always supposed to get the right information from the website of Food Safety Standards Authority of India, rather than just relying on negative messages which are passed on by the sometimes passed on by the press. However, press also plays a big role in creating positive opinion as well as creating awareness about various parts of the food safety regulations. Public perception, because this is also an important component in the food safety management, because over the years, Food Safety Standards Authority of India, which is the regulatory authority for the food safety regulation, has created a perception, positive perception among the minds of the people that packed foods or legal foods are safer than illegal foods or unpacked or unregulated foods. Because this is also very, very important component, that public perception of which is good, which is not good, is one of the driving forces of the management of risk. Bureaucratic imperatives also are there, law is there, economics is there, science is there, and ethics is there. All these components also influence the food safety regulation and food risk management in various ways. Science, I have already told you, the modern food safety regulation is science-based, and ethics and background of our country. For example, India is the only country where we call milk and milk products as the vegetarian, because green mark is given. Therefore, because it is by keeping the sentiments of the country, milk and milk products have been designated as vegetarian. But therefore, the green symbol do not just say it is vegetarian. Green symbol actually indicates on the food that either vegetarian or dairy-based products. The orange dot means anyway the product of animal origin. Therefore, this one slide I always like, because before we actually assess the risk or risk from a contaminant or an additive, you will not know exactly what is the risk which is proposed by that. Therefore, the orange or the purple line here, you will be seeing that the risk is just diffused. Therefore, when a specific risk assessment is conducted, you can see the the graph here. You can actually see that actual correct assessment of risk leads to the actual spread of estimates, with a small number of high risk and small number of low risk, and high number of actual correct assessment of risk actually happens here. Therefore, in the food safety outline, if you see, we all know why the food safety assessment is important. I have already talked. Why do we need to do the risk assessment? And we have to create the logic tree where events and fault tree is created. Description of value chains is also made. Proper field surveys for highlighting the importance of diagnostic tests is also done. Construct and run a risk model, because when you are talking about food safety risk assessment, many people always think that risk assessment a laboratory analysis. No, laboratory analysis and laboratory test are only one component of risk assessment, and it is actually a combination of a laboratory analysis or the science, statistical model, epidemiology, and economy, and much more than that. Therefore, how food safety risk assessment can be applied to improve the farm hygiene is also addressed in modern food regulation. Therefore, today, as per the food safety regulations, when a food business operator or a person who is dealing with the food processing, actually highest importance is given when he or she applies for either a registration or a license. There is something called Schedule 4 of Food Safety Standards Act or Food Safety Standards Regulation. As per the Schedule 4, the first license is given. What is Schedule 4? Schedule 4 is nothing but the responsibilities and duties of the food business operator with respect to hygiene and sanitation. Therefore, the actual importance what is given while giving the registration or licensing actually is his duties and responsibility with respect to hygiene and sanitation. Therefore, if you take the overall concept of risk assessment, it is basically database. It is not a, what do you call, a broad this thing. Okay, something is, because people always say that no, processed food is bad, unprocessed food is okay, packed food will have a lot of the additives, therefore we should not eat, unpacked food is better. Therefore, many times actually people go by their broad perceptions rather than scientific data. But however, risk assessment is not based on any of these the feelings, but it goes only by the scientific data which is available. Therefore, these scientific data actually drives the risk assessment, leading to an estimate. We call it as risk estimate, which helps us to make the risk management. Therefore, risk assessment is a process of identifying a hazard and estimating the risk represented by that hazard. Therefore, a framework could review and respond to food safety problems in a systematic, structured, scientific way in order to enhance the quality of decision making throughout the food chain. Please remember that the food safety assessment or food risk assessment starts with an administrator asking the scientists to do the food safety assessment, and finally all the science and technical aspects are finally bound into a simple information which can be conveyed to the government to say that yes, this particular additive can be allowed and this is the concentration in which it has to be allowed. Therefore, starting from a simple request by the government or the authority to the scientists, doing a complicated analysis based on the data and giving an output to government or the regulatory authority in the simple fashion that yes, allowed or not allowed, and if it is allowed, what is the concentration in which it is allowed. Therefore, when we are doing the risk analysis, the food safety policy is the one which actually drives the risk assessment, where we have risk assessment, risk management, and risk communication, which all these three things together makes the policy of food safety. Therefore, risk assessment assesses the risk management, risk assessment is done by the scientific fraternity, risk management is done by the regulatory fraternity in coordination with the scientific community, and the risk communication is the job of the government to, and also anyway, with the media, to convey the information in the right form to the consumers. Therefore, with all these three things together, exactly an agreed level of consumer protection is brought about. Please remember that when we are talking about food safety regulations, food safety regulation is done by your country, but not in isolation. Modern food safety regulation through Codex Alimentarius is done through a process of harmonization, where all the member countries are supposed to be, going by the guidances of the, Codex Alimentarius, and fixing the limit based on the requirement of the country. That means you are not going to just bring and adopt what are the guidances given by the codex. But however, your country, food safety regulator of your country, based on the input from the scientists, moderates and adjusts according to the requirement of the country. Therefore, risk analysis should be based on available scientific evidences, information on perceptions, costs, because why I'm saying cost is that each country decides on fixing the levels based on their ability, environmental aspects, cultural aspects, etc. Because again, cultural aspects also are something to do with the sensitivity of the population. Therefore, all scientific, perceptual aspects, cost aspect, environmental aspects, and cultural aspects also become important. Here, I just told you about, milk and milk products being a part of the vegetarian diet, which is gathered and analyzed according to the scientific principles to the extent possible. Therefore, when you do the risk analysis, you will actually be developing an estimate of the risks to human health and safety, because this is again a statistics. Identify and implement appropriate measures to control the risks, and communicate with stakeholders about the risks and the measures applied. Because therefore, you are actually assessing the risk, you are managing the risk, and you are communicating. Basically what I have told just a few slides before: risk assessment, risk management, and risk communications. Therefore, you are seeing that here, that in the risk assessment, you are supposed to identify the hazard. First and foremost, you should know what is causing the hazard, and once you know what is causing the harm, you are supposed to characterize the harm or the hazard. Then you are supposed to identify the exposure. Even though if there is a risk and you are not, your society or your population is not going to be exposed, you don't have to worry, because if there is a product which is used in an extremely small quantity on a daily basis, for example, you are talking about table salt. If you are talking about a staple like rice or wheat or rice flour or wheat flour, therefore we have highest sensitivity when we are allowing something on the rice when compared to what we are allowing in the salt. When we are allowing something in the milk, we are much more sensitive, because the milk is also the one which is used by the vulnerable population such as children. Therefore, based on who is using it, how much is being used, how much will be the exposure, then only a decision will be taken. Therefore, all these things actually together, put together to make the assessment of risk. Once the risk assessment is made by the scientific community, then comes the risk management. Therefore, in the risk management, the scientists or scientific population, because if you see the Food Safety Standards Authority of India's website, we have, there are many scientists groups called the scientific advisors, and from them, once the risk assessment is done, the management of the risk is done by keeping in mind the food safety regulation. Therefore, it will assess the policy alternatives, select and implement appropriate options, and once that decision is taken by the Food Safety Authority, then Food Safety Authority will take a decision to communicate in an appropriate fashion. Initially, risk management aspects will be put it in the Gazette, as these are the decisions taken. Once those things appear in the Gazette, scientific, the regulatory matters are finished. But once you do that, it has to be, this particular aspect has to be properly communicated. For that, various risk communication strategies are adopted, where actually interactive exchange of information and opinions happens. In fact, once the decision has been taken by the Food Safety Authority, it is once it is put in the Food Safety Authority website, and after the Gazette announcement, they actually once the decision is taken, 1 month or so is given for the objections by any of the organizations or even individual as such. Once all these comments are given within time, they will actually have a relook at the policy to find out whatever the policy which has been developed is accepted or not, and whether these, if there is any appropriate suggestions or the comments or the objections which are raised, will be rediscussed with the scientific community, the scientific panels, members of the scientific panels, and then only a decision is taken. In fact, I'm also the member of the scientific panel of sampling and analysis at Food Safety Authority. In one of the cases, when a decision was taken, it has come back several times before the final decision is taken, because once you put it for public comments, there will be either the concerns or the objections raised by various stakeholders. Once it is done, final Gazette notification is done, and then they will develop strategies for communicating it to the population or the stakeholders. Therefore, we talked about two words till now. One is risk, one is hazard. Are these same? Are they different? What is a risk? Risk is a function of probability of an adverse health effect and the severity of that effect, consequential to the hazards in the food. Therefore, it looks a big jamela to you when I read this, but if there is a small pin, which is because you must be knowing traditionally, they used to pack prasad and snacks in a plastic sachet, and they used to put a stapler pin. And now they don't do it anymore because it is illegal to use a safety pin, use a stapler pin in the food product, because Food Safety Authority has banned it. Why was it banned? Because these stapler pins are considered to be a physical hazard in the food. Therefore, that means an assessment of risk was done to find out whether it is really a hazard. Then they have made a policy that it is not allowed, and that is communicated to the people. Therefore, the food industry no longer uses that as a method to pack the food product. Therefore, if anybody still does it, that's illegal now. Therefore, even though now let us move one step forward. In the scientific panel where I am there, one more hazard keeps coming. Keeps coming in the meeting. That is stapler pins which are used in the tea bags. Just imagine, because now how slowly we are climbing up the ladder of the level of safety. The Food Safety Authority or regulators are sensitive to even the stapler pins which are used in a tea bag. The food safety regulators are also sensitive to small metal pieces which come in the tea powder in the process of tea making. These are considered to be a physical hazard. Then in addition to that, many other chemical or biological hazards which we will see a little bit later on. Therefore, these are nothing but any material which has a capacity to cause hazard, which is a physical one, chemical one, or a biological one. Therefore, any material which has the ability to give a risk. Therefore, risk, when it is taken at a next level, we call it as a hazard. Risk means probability of an effect. Hazard is the actual perceived risk in the form of biological, chemical, or a physical agent, or a combination or a condition of a food with the potential to cause an adverse health effect. Therefore, risk and hazard are the same, but risk, when it is perceived as a factor, becomes hazard. Therefore, you can see the one small cartoon here. On the left side, you can see a couple of people sunbathing. Okay, when they're sunbathing, there was an umbrella which is closed here. Somebody is directly, sleeping directly to the sun. And once the sun rays become stronger and stronger, what this person has done is he opened the umbrella. Therefore, he or she, I can see it's a she. She was more careful. Women are more careful, I think. She's more careful, and she has used this umbrella and protected herself. But the person who is here, who did not bring an umbrella, is exposing himself to the sun. Now you can see that that fellow is suffering from the extreme effect of the sun. But the hazard is, the risk is already there. Hazard is also there. But the lady on the right side has taken precaution to mitigate the hazard. That means she has assessed the risk, that she knows that there is a risk which is going to be there, and also she has managed the risk by bringing an umbrella, and she has opened it. Probably she has communicated to others also. Next time while coming, this person might come up with an umbrella. Yes, if I bring an umbrella, I will have I will have the safety here. Therefore, risk is equal to hazard hazard multiplied by exposure. Therefore, when you are, we know what is the hazard now, risk now. Risk is the hazard multiplied or as a factor of hazard and exposure. Just as a hazard is only a risk. Therefore, hazard is an agent with the potential to cause harm. Now you can see the picture of these three pictures, nothing to do with food anyway. On the left side, on the top, you will be seeing an actual crocodile. On the left side, you have preserved crocodile or a mummified crocodile. On the right side, you have a very small, tiny baby crocodile. if you see this A, B, and C, all these, when I did not, if I did not show this picture, I would have said A, B, C are the crocodiles. But now, when you see this, you know that yes, it is, but none of them can actually have the ability to cause except A, which is the real crocodile. Therefore, the actual hazard, when it has the capacity to expose you, only will become the which can actually become a risk. For example, here if you see, also we are talking about the vegetables here on the left, which are in an either hygienic or unhygienic conditions. In the middle, you will be seeing a food which is exposed to a fly. On the right side, you can see a vegetable with visible growth of fungus on the surface. Therefore, if you take the food in general, hazards in the food can be physical, chemical, or biological. All these three things can actually cause a risk, but they will be, they are causing different kinds of hazards. Intentionally, I have put three different colors for these three hazards. Physical contaminants are hazards, but physical contaminants can cause physical risk. Chemical contaminants can cause a risk starting from very simple to very severe, and also chemical components actually can be present at various levels. Starting, it can either remain the same till the end of the processing, or also it can decrease. But as far as biological contaminants, I have put it with red, because biological contaminants, especially microbiological contamination, is always marked as red, because this has the ability to magnify in a very, very short span of time. Therefore, when you do the risk assessment, inputs such as what kind of food, what kind of hazards, what kind of population groups intended, process models, consumption models, dose response models, as well as the health outcomes. All these data are taken into consideration when the risk assessment is done. When you are doing the risk assessment, number one, or the four most important one, is the identification of the hazard, characterization of the hazard, assessment of the exposure, and lastly but not least is the characterization of the risk, which happens based on the data which are available here. Therefore, it is not just, please remember that it is not just the hazard, but hazard versus the nature of the hazard, and also the exposure. I have already told hazard versus as a factor of exposure. Therefore, when you are doing the risk assessment can be quantitative, like yes and no, low or high, or many times, in many cases, it is actually quantitative. That is, for example, in case of the pin, we are saying it is yes or no, whether it is there or not there. And in case of, for example, the chemical contaminants and biological contaminants, it will be quantitative. Number of cases per million, 10 million, or annual cases. Therefore, and here, when we do this risk assessment, we always keep one thing in mind, that is disability adjusted life years is taken as the benchmark here. That is some of the years of potential life lost due to premature mortality, and the years of productive life lost due to disability. This is taken as the benchmark where a decision, where you are assessing actually the risk. Therefore, anyway, this part I will go quickly, technical aspects of it. Therefore, when you are doing the hazard identification and characterization, you will be interpreting or taking all the results from these into human, interpreting it to human toxicity data. Therefore, in the in vitro studies, using the using the organisms or cells or tissue preparations from laboratory animals to humans at various levels. In vivo studies, that is, we are talking about biochemical studies. In vivo studies in laboratory animals or humans. Identification of potential adverse effects, such as, for example, hazard identification. Then no experimental, please remember that no experimental species is an ideal model for humans, because they can only represent it. They cannot be taken as the virtual replacement of human studies. there is evidence that studies in animals generally provide an effective means for evaluating potential toxicity of substances in food. However, it is not the complete data. But today, anyway, we are one step further, because once the animal experiment, before the animal experimentation as well as after the animal experimentation, we are in a position to do in silico analysis, where we will be able to see many of the, or we will be able to do many of the work which we would not have been able to do in the past. Adequacy of design and conduct of the experimentation should preferably conducted as per good laboratory practice. Because anyway, Professor Mohanan is here, he will agree more with me, because many of the, in Food Safety Authority, when we are doing the risk assessment of many of the additives or even the contaminants, what happens is that we will look into all the papers available in that subject in the past. We do a literature survey. When we do the literature survey, we are many times actually in a fix whether to use a data or not, because many of the scientific studies which are done need not be useful for us to do a risk assessment, because each study is done in different conditions. Therefore, you will not be able to compare the two studies together. Finally, the Food Safety Authority is arm twisted to do the entire study again by spending the money, even though so much of literature is already available. Therefore, you all know this anyway, all the different levels of toxicity from acute to long-term toxicity studies, mutagenicity studies, carcinogenicity studies, teratogenicity studies, multi-generation studies. And with all these, you actually try to come and fix a level that is low observed adverse effect level, that is the least concentration in which you are able to cause any of the symptoms, or establishment of no observed adverse effect level. Once you do that, you will be you will be doing various studies, starting from exposure and to toxicity. And once you do these, you will be moving to the step of hazard characterization, where you will be actually seeing the dose levels, that is toxic effects. That is the ability of the chemical to cause the toxicity at certain concentration, but not the death or severe suffering. And once that is decided, then lower dose levels producing graded responses and no adverse effect at the lowest level. That means first you find out the LD50, LD100, you normally do LD50, and slowly come down where you are able to show the responses, but there is no severe effect, and finally come to the lowest level to cause these effects. And then you come to the point of departure. One of the standard point of departure is no observed adverse effect level in a species, because these are species specific. And this is considered as a reference point in the hazard identification. When you are doing the hazard characterization, again, dose response data, that is risk assessment, advice, quantitative, qualitative risks, and health-based guidance values such as acceptable daily intake, laboratory studies, then most importantly, NOAEL, as I've already told you, and many times you will be doing it in a very sensitive species. Then you move up the ladder with the various species. Finally, you come up with a safety uncertainty factor. Mostly it is 10 into 1000 units per mg/kg body weight. Therefore, please remember that when pharmacologically, Dr. Sham and Dr. Mohan will agree with me, when they are trying to give a chemical as a drug, you are accepting certain level of toxicities or adverse effect based on what level in which you are making the treatment. But with respect to food, please remember that always it is a thumb rule that there should be no health hazard whatsoever. Therefore, level is fixed for the additives called acceptable daily intake. That means if you are consuming this particular additive in such a concentration throughout your life, you should not have any perceivable health risk. But in case of contaminants, for example, heavy metals, mycotoxins, and so on and so forth, we always give another value which is almost similar to this as tolerable daily intake, or TDI, because there we are talking about the highest level with which no effect is there, therefore this level can be given. In case of TDI, that is in case of contaminants, we always try to give as little as possible, so that the we are allowing, not giving, we are allowing as little as possible, so that it can be there, it can be present in the food without causing health effect. Therefore, if you see it as a relationship between each other, ADI, NOEL and LOEL, therefore, LOEL, if it is around 600 mg per kg body weight, then NOEL, say it is 400 mg per kg body weight, then now we will be giving the safety factor of 100, and you come to the value of 40 milligram per kg body weight. Therefore, in the exposure assessment, we will be talking about food additives and their concentration data from the manufacturer, because manufacturer, when he applies, he gives it with all the data available with him when he applies for the permission. Food contaminant concentration from the surveillance and other sources, because in case of food surveillance, nobody will come for the approval of it, but always a food manufacturer will come and say that don't fix at too very low level, then all our consignments will get rejected. Therefore, in case of food additives, manufacturer will come out with the levels of safety, and in case of the food contaminants, it is the population surveillance data. Once the regulation is made, the food safety regulator will be looking into specific food containing the food additive or contaminant in the market. They will be looked into. Actual levels of food additives and contaminants from the foods are measured. Analytical data on the concentration of food additives or contaminants in the food may also be used more realistically to estimate the levels of food additives likely to be found in the diet which are consumed. In addition to that, monitoring and surveillance also is done continuously. Therefore, various approaches are used, such as food consumption data, which are population based, diet surveys, per capita consumption, which are normally useful, not generally useful for the food additives actually, but mostly for the contaminants. Household based data, and also individual data which will be collected. And recently also National Institute of Nutrition Hyderabad has done a total diet study to know the comparative exposures also. Therefore, I have a few more slides with which I will be able to finish in another 5 to 8 minutes. And this, just to break the continuity, I brought this. All things are poisons. Nothing is without poison. Only the dose makes a thing not a poison. Therefore, everything is poison, but how much you take determines. Therefore, our great Indian saying is also there. If even the amrutam becomes poison if you take too much of it. Therefore, the few slides on the risk management. If you see the general principles of risk management, a whole intention is protection of human health with the transparent decision and practices done by the regulator, and functional separation of risk assessment and risk management. Therefore, these cannot be done together because each one of these should not influence each other. That is the reason in the Food Safety Authority, the scientific panels are independent scientists and this food safety regulator is a different. Therefore, there should be a continuous process. And with that, actually, the food safety regulator will have various standards which are developed. That is, Food Safety Standards Act 2006, when it was implemented in 2011, has come out with the food safety standards regulations, which are nothing but the set of standards. These set of standards are done with various food categories, with as horizontal standards and vertical standards, and these horizontal standards are there specifically for contaminants, residues, packaging, as well as labeling horizontally. That means it cuts across all the food categories. Vertical standards are the standards for specific food categories. Biological hazards actually give the hygiene as well as microbiological standards. Chemical hazards, in food safety management, in the risk management, microbiological standards actually handle the risk management of biological hazards. For the chemical hazards, in case of food additives, permitted levels. In case of contaminants and residues, it is maximum residue limits. Veterinary drug residues also the same case, and the environmental contaminants tolerance limits, and principle of ALRA is used. This is a very important, as low as reasonably achievable. Many times people ask questions, why do you allow 10 ppb of the aflatoxin in this particular food? Why are you allowing certain amount of microbial colonies in the food? Why are you allowing certain amount of heavy metals in the food? This is because if you make everything as zero, all the foods what we consume becomes not conforming to standards, and all these things will get rejected. You are not supposed to allow them. Therefore, the food safety regulator step by step lower the limit, and side by side taking actions to lower these limits in the food products. Therefore, food safety hazards in foods, if you see, either they can be intentionally added, such as food additives and processing aids, where highest level of importance is given for the risk assessment. Second group is not intentionally added, such as metal contaminants, toxins, materials which come in contact with food, pesticide residues, veterinary medicines, hormones, which come actually at various stages of food, processing chain, and you have physical, chemical, and biological contaminants, which I've already told you. Therefore, if you, when you do the hazard characterization, you will be actually the qualitative and or quantitative evaluation of the nature of the adverse health effects associated with biological, chemical, and physical agents which may be present in the food. And for the chemical agents, it's a dose response assessment which will be performed. For biological and physical the dose response will be done based on the data obtainable. In the hazard characterization, we will be actually giving a description of the harm consequent to the exposure to the hazard, severity of harm, which is qualitative or quantitative. That means based on the data collected. For example, in case of a child getting a small injury, but it might look serious, but it is not really serious. But in case of an accident, and the child coming in front of a car, this is more severe. Therefore, how you perceive that particular data. Therefore, in case of microbiology, the hazard data we are talking about, what kind of sources you are talking about, what kind of symptoms you are talking about, because these are the literature which is already available. Therefore, in fact, even when we are doing the food safety regulation, it is called as risk-based importance. For example, milk, meat, poultry, and oil are a couple of the foods where highest amount of risk of microbiology is there. Therefore, these are the ones which are given highest importance when we are doing the food safety regulation. That is what is called as risk-based approach. Therefore, when we are doing the exposure assessment, we'll be searching, collecting, collating, analyzing, and summarizing all the data which are available to make a model for food consumption and exposure of the individuals to risk-related consumption of the food, incidence and prevalence of biological risk, residues of various contamination, and identification of the risks which are emerging. Please remember that when the food is permitted category wise. When you do category wise, please remember snack food is snack food, staple food is staple food. But however, please remember that Maggi is not considered as staple. Maggi is considered in the snack category. But there are households where Maggi is the breakfast, Maggi is the lunch, Maggi is the dinner. Therefore, I'm sorry, I'm not supposed to take the name of a brand, but a noodle can be all three. But noodle which is made at home is different in China when compared to instant noodles which we are consuming in many of the families. Therefore, but when the product is approved, it is approved as a snack product. When something is approved as a snack product, you are considering the exposure only with respect to certain percentage of the total daily intake, not as 100% daily intake. Please remember that. Therefore, what is how the exposure happens? One is proximity to hazard. Here you are seeing a cartoon here on the left that you are swimming next to a shark. Risk is very high, but only shark is there. You are not in the pond or you are not in the ocean near the shark. Then hazard is there, but no risk is there. Therefore, hazard proximity to the risk makes the hazard. Therefore, in case of food, consumption of foods containing the hazard, and how much of it you are consuming makes the exposure more important. Anyway, now you can see here on the left, you actually can see children who are not bothered about what is happening in the traffic next to them. A child crossing the highway, you can see here. Anyway, this one I'll skip. And therefore, in the hazard characterization, hazard identification, characterization, and finally making the risk assessment. I will talk a few words about the risk communication. Risk management is the most important one, where you evaluate the policy alternatives and then discuss or consult with the concerned parties. As I've already told you, it is discussed in the Food Safety Authority. There will be also representatives from the consumer organizations and farmer organization. Finally, it is put in the website of the Food Safety Authority for the comments of all the people. Therefore, then they consider the risk assessment and other factors relevant for protecting the consumer's health, promotion of fair trade and practice, and if needed, selecting appropriate prevention and control options. Therefore, in the risk management, you can say something like this. On the left down, you are seeing a school bus and children getting out. On the right corner, you are talking about a child crossing the highway. Therefore, if a policeman is there near, a policeman or woman here is there near when the school bus is coming, now protection the risk is managed. And the same thing is mother is taking the child with her when she is crossing the highway. Therefore, this is supposed to be the last slide of mine, where I'm putting it together here. As far as India is concerned, our Food Safety Authority, Food Safety Standards Authority of India is the one who is the food safety regulator, who is coordinating all the aspects, starting from making the regulation standards, as well as its implementation. And when you're doing that, the how FSSA deals with it is actually summarized in this one. You can see the risk assessment is done by them, where by using scientific panels and scientific committee, and risk management is also done by the food authority. Then risk communication is done in in coordination with the press and the information broadcasting ministry, and enforcement of food safety regulation is done by Food and Drug Administration of the states and union territories. And overall, it is the consumers and other interested parties are whose interest is kept in mind when all these things are done. In the bottommost, I have put a line there: National Codex Committee and the Codex Shadow Committee. That means this is component. This is part of food safety regulation. Food safety regulator, that is Food Safety Standards Authority of India. At the world level, we have the Codex Alimentarius, which is a world body, which is a guidance body, and the Food Safety Standards Authority is a member of that through a National Codex Committee, and there is something called Codex Shadow Committee, which helps in the harmonization of food safety regulation of India with the other countries. With with this, I would rest, and thank you very much for Dr. Mohanan, Dr. Nair, and all the organizers. I thank you very much for giving me an opportunity to discuss these aspects with you. Thank you very much, and I hope some of these messages carried through to the participants, and I am happy to answer any questions if they have. Thank you.
There is one question.
Yeah.
Professor, you can able to see here, or we can read it?
Yeah, you can read. I think that is better.
Okay. The first question is: there are regulations for assessment of packed food.
I'm not able to hear you well.
Okay. There are regulations for assessment of packed food. What are provisions for quality assessment of unprocessed food like vegetables that are many times stored, housed for many health risks arising from heavy metals and other contaminants?
Yeah, it's it's a very good question. I'll I think address it with a little bit of a one-line discussion before that. What happens is, when you're doing the food safety regulation, the there are two groups. Foods are categorized into two groups. One is the regulated foods, that is standardized foods, what we call, where the food either it is a processed food, or even the rice which is not processed also is in a regular packed food. Therefore, I will put them into two categories. One is packed food, one is not packed food. When we are talking about a packed food, there are standards for those. When we are talking about the openly sold products, for example vegetables, and agricultural producers, for example fruits, and so on and so forth, regulations don't apply there. What applies is the guidelines. For example, as per the Agriculture Ministry, guidelines are there that vegetables are not supposed to be grown in the nalas or contaminated water. In fact, In fact, this discussion came just 15 days, 6 months back in the FSSA in one of the committee meetings, where they were saying that a large number of vegetables are being contaminated with heavy metals. What to do? Therefore, FSSA actually has made a committee which is going through all the vegetables which are grown in different parts of the country to find out what is the baseline data with respect to that. However, with respect to these vegetables getting contaminated because they are being grown in an unhygienic place or they are not supposed to be grown, there is a much, quite a bit of clumsy issue which is coming into force. For example, if a vegetable is being sold by somebody on a cart which is contaminated, will you be able to arrest that person for that? No, because it is the consumer who has to make a decision what kind of vegetable where they are buying. You understand what I'm saying? But as per the government regulations, they are supposed to use only water which is supposed to be used for irrigation only for the growth of green leafy vegetables and vegetables. Therefore, there is as I've already told you, one concept that as low as reasonably achievable. Therefore, as per the government guidelines, if the manufacturer or the producer is following the guidelines, the heavy metals and chemicals coming in contact with these is very low. Yes, there are unethical practices done by some of the growers. However, regulating that to the finest thing will be very difficult for the government unless and otherwise there is proper awareness among the people who are growing and also proper awareness among the people who are consuming it. Hope I answered it in a half right, half wrong way.
Thank you, sir.
Yeah.
Thank you for giving us a very informative lecture, which made us aware about the possible hazards and risk in relation to food. Thank you for sharing your knowledge and have a nice day, sir.
Thank you.
Thank you, Professor Shetty. Thank you so much.
Thank you, Dr. Mohan. Now the closing ceremony is going to be followed by this, or?
You're going to be there. Be there.
Okay.
We are grateful to all experts who have come to share their knowledge in the 3-day webinar series on food, chemical and nanomaterials toxicity, jointly ventured by Sri Chitra Tirunal Institute for Medical Sciences and Technology, Central University of Kerala and Kerala Academy of Sciences. We hope everyone had a fruitful and rewarding exchange of knowledge during these 3 days. We greatly appreciate each and everyone for making this webinar series a most worthwhile experience. Please do feel free to share your experience with us. Now going over to the valedictory address. Now I invite Dr. Megha to compare the valedictory function.
A warm welcome to all. Now we are moving to the valedictory function. We request Professor G.M. Nair, Dr. Harikrishna Verma, and Professor Suresh Das kindly be online. With great respect, we invite the organizing secretary, Professor Sudha K, head of the department, School of Biological Sciences, Central University of Kerala, to welcome the gathering. Over to you, ma'am.
Okay.
Check here. Just check whether you are audible or not.
Hello, am I audible? Okay.
Yeah. Yes.
Okay. The chief guest of the day, Professor Suresh Das, former director, National Institute for Interdisciplinary Science and Technology, Emeritus Professor, Indian Institute of Science Education and Research, Trivandrum, respected Professor G.M. Nair, President, Kerala Academy of Sciences, Dr. Harikrishna Verma, Head, BMT Wing, Sri Chitra Tirunal Institute for Medical Sciences and Technology, Dr. P.V. Mohanan, Organizing Secretary, scientists from various institutions, invited guests, participants, colleagues, and my dear friends. A very good afternoon to all. In December 1959,
Richard Feynman gave a talk called "There is Plenty of Room at the Bottom" at an annual meeting of the American Physical Society at Caltech. In his famous lecture, he imagined a day when things could be miniaturized, when huge amounts of information could be encoded onto increasingly small spaces, and when machinery could be made considerably smaller and more compact. Now, more than half a century advanced, we can see the light about it as nanotechnology becomes the foundation of incredible industrial application in almost all aspects of life, from cosmetics to powerful computers. Although human exposure to nanoparticles has occurred throughout human history, it dramatically increased during the Industrial Revolution. The extensive human exposure to nanoparticles led to significant concern about the potential health and environmental risk, which paved the way to emergence of additional scientific disciplines, including nanotoxicology and nanomedicine. In this scenario, the international webinar series on food, chemical, and nanomaterial toxicity was significant in exchanging views considering the diverse aspects of nanotechnology. This was more evident with the participation of 12 invited speakers and more than 330 attendees from the countries including UK, US, France, UAE, Japan, China, and India. The sessions reviewed the fundamental toxicological concerns of nanomaterials, medicinal application of nanotechnology, stability concerns of nanomaterials, application of nanotechnology in water treatments, potential application and benefits of nanotechnology derived products. It's my pleasant duty to bid you all a genial welcome. We are profusely overjoyed to welcome our chief guest of the day, Professor Suresh Das, a stalwart scientist who immensely contributed in the field of chemical sciences. We welcome you, sir, to this valedictory function of this webinar series. We are privileged to have Professor G.M. Nair, President, Kerala Academy of Sciences, as an important part of this program. Sir, we cordially welcome you to this valedictory session. We are also delighted to have Dr. Harikrishna Verma, Head, BMT Wing, Sri Chitra Tirunal Institute for Medical Science and Technology, to address the closing ceremony. We warmly welcome you, sir, to this valedictory function. Also, I am very happy to welcome the speaker, Pratap Kumar Shetty, Senior Professor, Pondicherry University. Welcome you, sir, to this valedictory function. As a custom, I also welcome Dr. P.V. Mohanan, Organizing Secretary, Sri Chitra Tirunal Institute for Medical Sciences, to this valedictory function. And I warmly welcome all the scientists from all 3 institutions to this valedictory function. As we know, attendees are the key component of any program. It's my pleasure to welcome all the participants to this closing ceremony. Once again, on behalf of the committee, I welcome each and everyone present here to this valedictory function of the 3-day international webinar series on food, chemical, and nanomaterial toxicity. Thank you one and all.
Thank you, ma'am. With immense pleasure, we invite Professor G.M. Nair, President, Kerala Academy of Sciences, to give presidential address. Over to you, sir.
Thank you. A very good afternoon to all of you. It's indeed a great pleasure for me to be there in this valedictory function, closing ceremony of the 3-day webinar. Kerala Academy of the Sciences has been a partner to this program, but the Academy did not do much in the organization part. We have been witnessing this, it was completely organized by Professor Dr. Mohanan and Professor Sudha. I really take this opportunity to congratulate Dr. Mohanan and Dr. Sudha for doing it great. It was well organized and the lectures were very good. For me, I didn't have much to do except to listen to the 12 speakers. For the 3 days, I've been listening to the 12 speakers, learned a lot. My knowledge bank has been augmented in such a way that it's fully loaded now. I did not miss any of the lectures. I was throughout hearing all of them, starting from, the talk by Jennifer, ending with Dr. Pratap Shetty. This is something great. All that begins well will end well. Started with the food toxicology program by Jennifer, and ended with the national level top scientist in food safety and technology, Dr. Pratap Shetty. And this shows that, and then the program, the 12 lectures that we have listened, out of the 12 lectures, 7 lectures were dedicated to nanotechnology and nanoscience. That shows the importance of nanoscience, nanomaterials, especially in the healthcare sector, in all the other activities which refers to the human beings. Most of them have discussed very well about the toxicity that normally is there for the nanomaterials, but then obviously they are a blessing that you could deliver your drug anywhere in the part of the body to the diseased regions of the body. The advantage would be that there would be very less of toxicity provided you take care of it, because anything that you use foreign material to the body will have its implications. So the question is that when you use a lot of nanomaterials in the body for all these treatments, or maybe for killing the cancer cells, they all get accumulated in the body. I was extremely happy to listen to Dr. Shakti Kumar from Japan, who I was not knowing that term, therapeutic diagnosis. It's called as theragnosis, where nanomaterial being used becomes a therapeutic material as well as it could be used for diagnosis. This is very important because, especially in cancer therapy, one of the major problems that affects, that makes its visibility is that the cancer cannot be detected at the early stage. Only at a later stage it is being detected. So if you can be diagnosed, if there are specific things on the cell surface of a cancerous, just a progressive cancer cell, probably that would be a great boon. And for that matter, I was very happy to listen to Dr. Shakti Kumar, and all the others have described about the various kinds of nanomaterials being used, their toxicity levels. And Neetu Singh from IIT Delhi, she talked about the mitigation levels that they are taking up. That also is very important because mitigating the effects, nullifying the toxic effects, because as I said, that if there is a kind of magnification that is occurring in the body, and you have a lot of magnetic particles and nanomaterial particles getting in the body is to be seen. I was also very happy to listen to Pratima Solanki today on biosciences biomarkers today morning, and it's also something which is an eye-opener because biosensors and biomarkers are very important. Biomarkers, for that matter, would be very important. Whether there could be identified biomarkers in the blood, which could speak about a cancerous event that's going to happen to your body, or maybe that there is a risk for cardiac problems. And if all these are there, I'm sure it would be great. Among the other speakers, I was very happy that when Dr. Jennifer had used a zebrafish as a model system for assessing the toxicity, especially on a very potent herbicide being used, which would be available in the fruits and the food that we take, the atrazine. she has assessed and all those that is happening. Dr. Dev Smita, they've had discussed about using Drosophila as material to assess, the impact of certain natural drugs, especially that artemisinin, which she said is something great. Artemisinin is a newly conducted natural product from Artemisia, and this drug discovery has paved way to a Nobel Prize in 2015. Yu To from Japan had got Nobel Prize just for discovering this in 2015 in physiology and medicine. So just imagine about this. We have been relying on quinolodine alcohol substances, which most of those malarial parasites have developed resistance. So it has been again a great breakthrough in identifying this, and Dr. Dev Smita has been using this for controlling cancer. That's again a great thing. I was also happy to listen to Dr. Vikash, learned a lot about the clinical trials being conducted, the phases of clinical trials, and all those activities. And connected with that, we had Dr. Sham Sharma, speaking about the GLP and GMP in pharmaceutical development and programs. I was also, as I said, that it was ended up with Dr. Pratap Shetty, a good friend of mine, because I read food is very important, because if you have a good food intake, your diseases, lifestyle diseases especially, will be less, cancerous incidence will be less, because as I said, that thy food is thy medicine. If your food can be medicine, that would be the great goal. I said in my introductory inaugural speech in the ceremony that the incidence of lifestyle diseases and all the other elements have come down, because for the past 8 months we have been confined to home and eating only home food, and no junk food has been allowed to get into our body, and that's again a positive sign for us. And for that matter, the disease load, the patient load in many of the hospitals have come down, bad for them, but good for us, because there are no problems associated. So I'm happy that the 3-day webinar is being organized in such a wonderful way, with all valuable speakers giving their in-depth knowledge on the subject, and it has covered almost all area of the 3 areas being projected: the food, the chemicals in the nature, in the environment, as well as on the nanomaterial toxicity. I congratulate once again Dr. Mohanan and Dr. Sudha for making this great seminar. I'm sure that you are all enriched with valuable knowledge, and when you go home, also you don't go home because this is all online platform. But I thought maybe that if it's a physical meeting, that you all go home, back home after valedictory. And I'm sure that you don't have much of patience to listen to people talking, giving lengthy lectures during valedictory, and people glued to the computer only when there are some awards to be given, to be announced. So maybe that valedictory will be full if there are prospective people who are going to get awards and recognition. But in this case, there are nothing except that I praise all the 12 speakers. Dr. Mohanan and Dr. Sudha has chosen them rightly, and I'm sure this meeting has been a grand success in terms of its content as well as the way it is being conducted. Thank you very much, and I'm waiting for listening to Dr. Suresh Das, who has been my boss in CST, Kerala State Council for Science, Technology and Environment, and a scientist whom I respect a lot. So maybe with that, I would conclude my presidential address, and I thank you all very much for being with us to organize this. And though I didn't do much, but I'm the gainer in terms of the knowledge I've gained. Thank you all very much.
Now, with great honor, we invite our head, Dr. Harikrishna Verma, BMT Wing, Sri Chitra Tirunal Institute for Medical Science and Technology, Trivandrum, to address the gathering. Over to you, sir.
Unmute.
Is it audible?
Yeah. Yeah.
Yeah.
Audible.
Yeah. Very good afternoon, and Professor Suresh Das sir, Dr. G.M. Nair sir, Dr. Shetty, the organizers, my dear students. The International Web Series on Food Chemical and Nanomaterial Toxicity, organized by Sri Chitradharana Institute for Medical Science and Technology and Central University of Kasaragod, in association with the Kerala Academy of Science, opened a new horizon of opportunity to understand the current research trends in the frontier area of nanoscience, technology, and their practical applications. There were about 12 eminent speakers, renowned speakers from leading institutes like Purdue University, USA, University of Houston, and TGH Chandigarh, University of Lorraine from France, from Toyo University, Japan, and IIT Delhi, NIPER and NICER, and from Pondicherry also, which shows the importance of this topic, as well as these very eminent speakers to spend their valuable time to share this interesting and novel research in understanding the real toxicology aspects of nanomaterials. The brainstorming sessions ignited new research possibilities in different levels of strategies of biomedical research as well. The lectures cover different toxicological aspects of nanomaterials, pharmaceutical products, and natural products. The deeper insight toward the importance of zebrafish and the fruit fly as model in the biological system, and how they are currently using for the nanomaterial assessment also, because these are the model systems which have been used over the years. The chemical toxicity of nanomaterials also the knowledge about the other current and a lot of natural novel possibilities, like in the hyaluronic acid based nano technologies for the theranostic based cancer therapy. The information about the development of one-dimensional semiconductors in clinical applications, as well as the cardiovascular safety assessment of these chemical entities were also highlighted. And I remember when there was an assessment and understanding of the DST project evaluation, there was a lot of interesting topic long before. This is the story about 10 years back when they understanding the bhasma of our traditional medicine. But at that time, there were a lot of suggestions were putting across to how to evaluate the safety aspect. Now, after 10 years, I'm 100% sure. Now we have improved a lot our understanding and the technologies to understand this safety aspect of such traditional medicine, and going to be real worth, and the people will also really study more scientifically understanding some of the traditional medicines as well. So I just wanted to thank all the eminent speakers for spending their time, and also I thank all the participants, especially students, and special thanks to the organizers, especially Dr. Mohanan and Dr. Sudha, for making this type of large gathering and people, and so much people are attending. And I could not completely follow some of the sessions, but like even the last session where Dr. Shetty was clearly mentioning about the food safety and the hazards and risk and other things, so informative, well prepared, and this will be an asset for the coming days. And I am sure our understanding is really improved with this one. And once again, congratulate the organizers, and thank you for all for attending. Thank you very much.
Thank you so much, sir. Now we are truly delighted to welcome Professor Suresh Das, former Executive Vice President of the Kerala State Council for Science, Technology and Environment, and the former Director of the National Institute for Interdisciplinary Science and Technology, CSIR Trivandrum. Currently, he is serving as the Emeritus Professor at Indian Institute of Science, Education and Research, Trivandrum. He established Photochemistry Research Unit at National Institute for Interdisciplinary Science and Technology, Trivandrum, and there he has instrumental in establishing a leading group, and state-of-the-art facilities for conducting research on basic and applied aspects of photochemistry. He is a fellow of the Indian National Science Academy, New Delhi, and the Indian Academy of Science, Bangalore. His research interests are in the areas of photoresponsive liquid crystals, near-infrared sclerine-based dyes, organic solid-state luminescent materials, and their self-assembly. On behalf, with great honor, we welcome Dr. Suresh Das to deliver the valedictory address. Over to you, sir.
Thank you very much, and welcome to all of you. I would especially like to thank Dr. G.M. Nair for inviting me to this valedictory function. It's a pleasure to be here. I have also heard a few of these talks, and I have to congratulate the organizers, Dr. Sudha, Dr. Mohanan, Dr. G.M. Nair, and all the others for the wonderful program, actually. As Dr. G.M. Nair rightly summarized, there have been some very exciting talks in this program. Of course, I have not much to do with toxicology, and in fact, when Dr. G.M. Nair invited me, I had mentioned that my expertise in this area is very limited. But you know how it is with Professor or Dr. G.M. Nair. It is very difficult to refuse a person like him. He is very good at arm twisting people into taking up. And so I am, of course, but it's still a very great pleasure to be here. He had also wanted me to make a presentation. So I have prepared a small presentation. Of course, it has nothing to do with toxicology. And I hope that this will be of interest. I thought I would share with you some of my thoughts on what it means to be a scientist and how scientists should be interacting with society. So if you, with your permission, I will just go on to my presentation. It says that the host has disabled my screen sharing. Will I be able to share my screen?
Yeah, yes, sir.
Yeah, can you see my screen?
Yes.
Okay. Yeah. Okay. So so I have made a small presentation, and I thought what I will do today is to share some thoughts on the roles of a scientist in society. In the last few days, you would have heard some really exciting and fantastic talks by experts in their areas on various aspects of food, chemical, and nanomaterial toxicity. And you would have seen how much of dedication has gone into their research, how much of sincerity and passion goes into that research. But very often, how much of that actually, and what kind of impact do these as scientists have on society? The reason I thought about this was very often when you go to places and like when you fill out your form, and there is an area which says profession, and we fill out profession scientist, And on occasions when we have gone to visit friends, and when the child there asks, "What do you do?" and I say, "You say you're a scientist," they get so excited. They clap their hands and they say, "You're a scientist." So basically what I wanted to say was that when the society thinks of scientists, this is what comes into their mind. This is what the impression is when immediately you say that you're a scientist. This is the image that they get in their mind. So every time we claim that we are scientists, when we call our scientists, we are actually laying claim to a very great legacy. And therefore, this is something that we cannot take lightly. When you see how scientists impact society, I just the reason we ourselves don't take ourselves as seriously, I think, as we should. And one of the reasons is that how we approach our profession. I just would like to share my thoughts on that. You know, when you think about scientists, you can ask three questions. What is it that scientists do? And then, of course, that naturally leads to how it is that they do their science. And then finally, the question, why we do all this, okay? So in order to, mention the importance of these three questions, I will take an example of a hypothetical scientist as to, so what we do, how we do, and why we do, okay? Take the hypothetical scientist, let us say a cancer researcher. And what does a cancer researcher do? Okay. Let us say that he is focusing on drug discovery, and that is what he does. How does he do it? We saw several excellent talks, even one today this morning. So on developing sensors, or you can think of in drug discovery, you think of developing a lead discovery molecules. You can do this in silico, you can make chemicals, then you can study their medicinal chemistry, and then you can do the efficacy and safety testing in animals, and finally clinical studies. And if it is successful, you have discovered a drug. So this is how they do it. But the question which is most important in this is, so we are going from what to how and why. And what I feel is more important is that we should actually begin with the why. It should be the other way around, not on what we do. We say that this is what we do drug discovery, this is how we do it, and that is why it is important. But in actual aspect, we should go going the other way around. The first question that we need to ask of whatever we are doing is, why are we doing this? And the what and how should subsequently come out of that. And I will elaborate a little bit. Say, for example, the why of the cancer research is awards and recognition. Then, of course, the focus will be in the early part of research, leading to some discovery of some fantastic molecules. If on the other, the why is wealth generation, the focus will be on the latter half. But let us say that the why of that scientist is fighting cancer in society. Then the entire approach of this person will be different, because he could be working on any other areas. But we know that cancer is not going to be cured only by this molecule. There are so many aspects. So such a person will be involved in many other aspects and the larger picture of how to mitigate cancer in society. Just like I have given here an example of the Union for International Cancer Control. So you can see that this would have been, such a society would have been started by people who are interested and have a larger picture in mind. Okay, so scientists who start with a bigger why, something that is beyond them, tend to take a larger picture, and therefore the science becomes, a total approach to that science becomes holistic. And that is what sometimes very often is missing. So we are, as scientists, we get so closely, our nose is so close to the grindstone that we don't see the larger picture. And so in order to do that, I think it is very important that we always begin with why we are doing what we do in the first place. Okay, so, and this is important because, I just wanted to help, because what it is, if you really look at in society, we are listening to a lot of people, and we hear in the debates and discussion, but how much of that debate and discussion is actually driven by scientists? What is the influence that scientists have in society in defining the agenda that is to be discussed? Okay, so what we call, for scientists, this is a circle of influence. Let us say, going back to the cancer researcher, this circle of influence is actually dealing with molecules, medicinal chemistry. That is where his expertise is. But if he is also interested in the larger picture, then you see that he also has a circle of concern. So as long as, of course, in order to be a good scientist, like in all the presentations that we saw in the last 2 days, you have to spend majority of your time on your circle of influence. That is where your core expertise is. But at the same time, you have to have this larger circle of concern in your area of interest. And like in this case, I have mentioned about people getting involved in other activities in society in order to mitigate cancer, other than just being concerned about your own research, because that is the larger picture that you have. Because when that happens, your circle of influence will grow, and so does the circle of control. And that is how the larger influence of scientists and science into society can happen. Okay, so I have a very interesting search I did for, on what people think about scientists. This was a question that was asked in Australia. People were asked to list the different types of people were given, different professions were given, and they were asked how do you trust, how untrustworthy or untrustworthy are people in this profession? And they were asked to scale them from 1 to 5. With 1 being the most trustworthy and 5 being the least untrustworthy. So in Australia, of course, doctors were considered to be most trustworthy, but you can see scientists rank fairly up in this. Whereas when you look at in the bottom, you can see, pollsters, politicians, government ministers, bankers, and journalists. They rank fairly down in this. And so the doctors, as I said, in Australia were slightly ahead of scientists. And if you look globally, you will see that generally people have a lot of trust on scientists. Okay, let us take another country, the UK, for example. It's interestingly, nurses are trusted most, and doctors. But you can see somewhere here, fairly on the top, the term scientist still exists. But this place is very common. It is almost exactly the same as what you see in the previous case in Australia. And it's the same thing if you take in the US. Okay, here also it is military, which is most trusted, but then you have medical scientists and scientists fairly on top. Okay, the reason I did, of course, we don't have a survey like this. I couldn't find one in our country, but I'm sure that it put in very much differently. But what is interesting here is that you can see how journalists, politicians, and others are perceived by society. In spite of the fact that scientists enlist such a large trust from society, my question is, how much are we actually impacting the way society is run? Okay, so because it's when I talked about the circle of concern. Now the circle of concern is important, but it's also the nature or the quality of the circle of concern is also important. Can the circle of concern be dealing with personal life of celebrities, social media discussion, politics in other countries, or religious differences? That is high decibel, low value. Or can the circle of concern deal with social development, health, agriculture, other important topics like economic development, low decibel, high value discussions? So the quality of that society depends on the nature of discussions it focuses on. Okay, what kind of questions are most frequently asked and discussed in a society? And what topics are discussed? Okay, and who decides what topics are decided? The top half of the trusted people, or is it the bottom half of the society which are less trusted? And you can see very often, very clearly, who drives the agenda for discussions in society. And how can scientists strive to say that we have to make more of an impact in society by continuously getting the discussions and getting the arguments in the areas where societal development and factors that are important to the development of our society become important. It is that which leads a country to become a developed country. So that is why I said, as scientists, when we make the claim that we are scientists, we have to take this statement a little bit more seriously. And we have, as I said, to see the larger picture and not just look at exactly, as I said, 80 to 90% of your effort has to go into your circle of influence where you have to develop the expertise. But the other 10, maybe 20% has to feature in your circle of concern. Then only you get the larger picture and have a holistic view about how research is done. So clearly scientists have an important role in directing the nature of debates in any society. And so, so that means this is what I call a virtuous cycle. That means this takes a society upwards. You build awareness, you organize demand, you focus on resources, you update policy and planning, enforce and adopt solutions. I have borrowed this from the World Resources Institute. And you can see, so if we are in this circle, society develops. But there will always be a tendency for those who control the politics and market to confuse the issue, bring about filters, and through fear mongering, distract our attention, or use through scapegoat politics or ineffective laws or short-lived results. So how can we enforce the system as scientists, that we ensure that the society stays within this so-called virtuous cycle that leads actually to development? Okay, so I will therefore come back to our 3-day conference that we have had, which was so excellently organized. We had some excellent papers on basic research, you had applications, you had interventions, and that is what I call the core circle of influence of people, researchers who are working on toxicity and food toxicity. And just the last talk was also so excellent on all aspects of food safety. And also we had the earlier talks on cancer detection and so on. So you can see that and some of this research will of course find, the basic research also will find itself into society, and we know that the whole process takes time. Okay, but at the same time, in an area, a topic which relates to toxicity, there are also larger implications to society. And where can toxicity be more important than cleaner, than creating the why should be that what the why for this research, of course, is that we are trying to create a cleaner, healthier, and less toxic world. And what could be more important than clean air, clean water, and clean food? So how much of those who are actually working on toxicology actually go out, because you're experts on toxicology, you're experts on how toxins in food make an impact. What are how much of an activist are you in making sure that society is on the right path on improving these things over and above just what you do within the laboratory in case of air purification or water purification? Okay, so you can see how important this is. Death from air pollution. We can see India ranks fairly high in the world. And if you look the top 10 polluted cities in the world, so we have at least 5 or 6 within these. And today it could be Delhi, and tomorrow it could be Trivandrum, where I sit. So these are issues that are important. And how do we get the societies to start discussing the importance of these issues? Of course, there has been a national clean air program that has been launched. Substantial funding is available, but there are lacunae, lack of legally binding targets, so on. So how do we ensure using this kind of circle, and how do people working on various aspects of toxicology, just like I mentioned about the societies that help to prevent cancer within society, how can the scientists organize so that the priorities are kept in check, and that we make the governments accountable and focusing on improving these kinds of systems? So that is when we go into that so-called 10 to 20% of your circle of concern, and ensure that the society actually benefits from the scientific knowledge that we develop within the laboratory. Okay, I don't, of course, want to leave that. So if you really look at it, and there are many scientists, of course, who do that. And I just thought I will conclude with a few initiatives around the world on how this is done, taking specifically the problem of cleaner air, for example. Here, this is the Friends of Earth in UK. What they have done is they have started an initiative where they let citizens test their own quality. So this brings awareness. So societies actually start measuring their own air, and that gets them interested, and on the question and the importance of clean air. This is one way of getting into society and acting upon your core interest, your passion, which is the sciences that is on toxicology. There is all over the world we are seeing some fantastic efforts in trying to bring about cleaner air through urban forests. These are also areas which many of us would be able to contribute in. This is innovations are other ways in which we contribute. This is an innovation from Karnataka, where people are provided with a small adapter which is kept at the end of your vehicular emission, and then you drop off that periodically to the company, and they derive ink from the carbon that is collected. So a bottle of ink like this is approximately capturing about 19.6 hours of a diesel car pollution. Okay, so that is again an initiative. So this is how we extend and into society, insisting that governments have cleaner vehicles, driving that into activity, driving governments and societies in that direction. And even, of course, this is not, it doesn't, all of it doesn't have to be that fanciful. Even this is a very good initiative in Bangalore on Exhale, Project Exhale, where you are making sure that the health of people in dwelling in remote areas and in areas where fuels are not available, that their health is also not not affected by bringing about more efficient stuff. So nothing is too big or too important once you realize that this is your area of interest and that is your area of concern. So I thought I will share these things with you because very often as scientists we get too locked up in our laboratory, in our research, in our area of interest, that we forget what we are and why we are in this profession in the first place. So I thought with these few ideas, I hope, I know this is not fully related with what you have been hearing for the last 2-3 days, but I thought I will share with you. And once again, thank you, Dr. G.M. Nair, for your invitation. And I congratulate all the organizers for the really wonderful lectures that we have had. This would educate our youngsters, and I hope that they will take the lead. And while doing their fantastic research in the laboratory, I hope that they will also keep in the mind why they actually got into this profession in the first place. And that is to create a better world in whichever area of research that we are, we have selected of our choice. So with this, I thank all of you and thank you for your attention.
Thank you, sir, for your thought-provoking presentation and enlightening words. Now, with great respect, we invite our organizing secretary, Dr. P.V. Mohanan, Senior Scientist and Head, Division of Toxicology, Sri Chitra Tirunal Institute for Medical Sciences and Technology, to express the word of thanks.
Good afternoon, all. Professor Suresh, sir, your talk or your presentation was excellent, like better than a toxicologist. It's very informative. I never see such a slides you're focusing in comparison with the global thing and different aspects of means the professional life and all. When you compare to the scientists, to doctors, to nurses, it's very informative. This is the first time I'm seeing such a slides. Very, very. Thank you so much for your such a wonderful presentation and information to us.
Thank you.
Okay, sir. Then regarding my job, like the vote of thanks, respected Chief Guest, Professor Suresh Das, sir, Professor Nair, sir, Dr. Harish Verma, our Head Biomedical Technology Wing, Professor Sudha, and my dear friend Shetty, Professor Shetty. And the scientific deliberation have come to an end today after 3 days of constructive discussions and interaction with multidisciplinary scientists. Today we have Professor Suresh Das, the former director, NIST, and former executive vice president of the Kerala State Council for Science, Technology and Environment is with us. In spite of his busy schedule, he accepted our invitation to deliver the valedictory address. Sir, on behalf of the organizing committee and on my own behalf, I wholeheartedly thank you, sir. Thank you so much, sir. I'm greatly thankful to Professor G.M. Nair, President, Kerala Academy of Sciences, and the chairman of the organizing committee and head Biomedical Technology Wing, Sri Chitra Tirunal Institute for Medical Science and Technology, Dr. P.K. P.R. Harikrishna Verma for their inspiring guidance and encouragement for the success of this webinar series. On behalf of the organizing committee and on my own behalf, please accept our sincere thanks, sir. Professor Shetty, please accept our sincere thanks for participating in this webinar series. My special thanks to Professor Sudha, Central University of Kerala, for her outstanding support for organizing this event. Once again, I thank you all for your kind participation in the international webinar series on food, chemical, and nanomaterial toxicity. Thank you all. Thanks. And please be with us for the national anthem.
पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य जय हे जय हे जय हे जय 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 हे.
Thank you, sir. Thank you so much.
Thank you, sir. Suresh Das, sir, thank you. Hello, sir. How are you? Suresh Das, sir. Yeah.
Yeah, how are you?
Yeah, fine,
Nice to
sir.
see you.
Fine,
Very nice
fine.
program I saw.
Yeah.
You had very good speakers. You
Yes,
can
sir.
hear me?
Yeah, sir.
Yeah, so very good speakers. In fact, enjoyed some of the talks, and I'm sure it would have benefited a lot of people. How are things there?
Yes, sir. Everything is good. Yeah.
Yeah, good. Yeah. How's your daughter doing? She's here, I think, Isa.
Yeah, yes, sir. Yeah, with
Professor
Dr. Mahesh. Yeah.
Yes.
Yeah, I asked her to meet you, but she said that sir always busy and
No,
running.
like
Yeah.
that. Yeah. Okay, I will. GM
Okay,
Nair
sir.
is calling me.
Okay.
I will leave you.
Okay, sir.
Thank you. Bye.
Thank you, sir. Thank you.
Bye.
Dr. Sudha, thank you so much. Dr. Sudha. Dr. Sudha. Okay. Thank you.
Hello.
Thank you.
I think from my part, I could, I have done as you expected, right?
Yes.
Mohana?
Yes.
What you have entrusted to me, that I could perform like that with the support of my students. Okay, my students, and I have so many students here with their immense support, and also I have a student. Actually, even though I didn't teach him, but he is my student, and so I thank my students, and also I thank Dr. Mohanan and your students were very brilliant research scholars for having given this great opportunity to us to collaborate, to join with your institution and join with your laboratory to conduct this wonderful seminar, especially the, the topic which is currently very important. So thank you so much. I hope that this webinar would pave the way for further collaboration, and maybe having with the the common interest, the area of common interest, and also the area of the interest of the society. So thank you so much, Mohanan. I congratulate you, and I feel I convey my hearty feelings to you for such a wonderful event we have conducted together. Definitely, I think this is a kind of a startup for our future endeavors, maybe in the area of research. Thank you. Thank you. So convey my regards to your students. I hope that our students will come over there and exploit. I mean, give you please give the opportunity to to extend you please extend your facility. I mean the equipment or whatever is available with you to our students who are also very interested to do something, especially by using some sophisticated instruments. I know that your laboratory is such a, with so many sophisticated equipment. But in Kerala University, in Central University of Kerala, we are developing. So that's why, but the current students, they need such a support, especially from your institution, especially my great friend, my very great friend, Dr. Mohanan, and also from and your students. So
We will
please.
support. We will support. Okay. We are welcome to all, especially all central students. Okay.
Pardon me.
Thank you so much. Bye.
Yeah.
Okay. Okay. Bye. Now it's time for lunch, no?
So thank you. Thank you.